For more than 3 billion years, life on Earth existed in forms so simple that you would not have recognized them as life at all. The oceans were filled with single-celled organisms, bacteria and archaea drifting in the currents, invisible to any eye that might have watched them. The seafloor was carpeted with microbial mats, thick layers of cyanobacteria that stretched across the shallows like endless green quilts, absorbing sunlight and releasing oxygen in a slow, patient rhythm that had continued for eons. There were no animals, no movement beyond the gentle pulse of the tides, no eyes, no shells, no predators stalking through the water. Life was abundant, yes, but it was monotonous. A planetary garden tended by microscopic beings that knew nothing of the complexity that would one day emerge from their descendants. This was the world as it had been for the vast majority of Earth's history. A world where evolution operated at a glacial pace, where change accumulated so slowly that 1 million years might pass with barely a noticeable difference. The planet was alive, thriving even, but it was locked in a kind of biological stasis, as though life had reached a comfortable equilibrium and saw no reason to push beyond it. If you could have visited those ancient shores, walking along beaches that have long since been ground to dust and reformed into mountains, you might have assumed this was all life would ever be. And then, 541 million years ago, something changed. Within a span of roughly 20 million years, a duration that seems impossibly long to us but registers as barely a moment in geological time, the oceans underwent a transformation so dramatic that scientists still struggle to fully explain it. The fossil record, which had been sparse and dominated by simple, soft-bodied organisms, suddenly exploded with diversity. Creatures with hard shells appeared, their bodies armored in calcium carbonate. Trilobites, arthropods with segmented bodies and compound eyes containing hundreds of individual lenses, began crawling across the sea floor. Predators emerged, some reaching lengths of 1 meter or more, hunting with sophisticated vision and grasping appendages that could tear apart prey. The first chordates, small swimming animals with a flexible rod running down their backs, appeared in the water column, and from this humble beginning would eventually arise every vertebrate on Earth, from fish to amphibians to reptiles to mammals to us. This event, which we now call the Cambrian Explosion, was not gradual. It was not the slow, steady progression that Charles Darwin had imagined when he formulated his theory of evolution by natural selection. It was an eruption of complexity, a biological Big Bang that produced virtually every major animal body plan we see in the modern world. And it happened so quickly, at least by the standards of deep time, that it seemed to violate the very principles of evolutionary theory. Darwin himself was acutely aware of this problem, and he considered it one of the gravest objections to his work. If evolution proceeded through countless small modifications accumulating over vast stretches of time, why did the fossil record show such a sudden appearance of complex life? Where were the transitional forms, the intermediate steps that should have been preserved in the rocks? For over a century, paleontologists searched for answers. They excavated fossil sites in remote corners of the world, places where ancient seabeds had been lifted into mountains and exposed by erosion. They developed new techniques for analyzing the chemistry of ancient rocks, measuring oxygen levels and carbon isotopes to reconstruct the environment of the Cambrian seas. They studied the genetics of modern animals, looking for clues about when key innovations first appeared. And slowly, painstakingly, a picture began to emerge. The Cambrian explosion was not a random accident, not a stroke of cosmic luck. It was the result of a perfect storm of conditions, a convergence of environmental changes, evolutionary innovations, and ecological pressures that had been building for hundreds of millions of years. The planet had been preparing for this moment, assembling the pieces. And when everything finally aligned, life seized the opportunity with a creativity and speed that would never be matched again. Tonight, we are going to explore that moment. We are going to trace the evidence preserved in rocks that have survived for 500 million years, examine fossils that capture the strange and wonderful creatures of the Cambrian seas in exquisite detail, and piece together the conditions that made this explosion possible. We will look at the oxygen levels rising in the atmosphere, the chemistry of the oceans changing in ways that allowed animals to build shells and skeletons for the first time, and the predator-prey arms race that drove innovation at a pace evolution had never seen before. We will meet the trilobites and the anomalocarids, the tiny chordates and the bizarre soft-bodied creatures that defied easy classification. And we will come to understand why this single chapter in Earth's history, this 20 million year window, matters so profoundly to everything that came after, including us. Because every complex animal alive today traces its lineage back to the Cambrian. Every fish in the ocean, every insect in the air, every mammal on land descended from the creatures that emerged during this explosion. The body plans that evolved then, the basic blueprints for how to build an animal with eyes and limbs and a nervous system, have been refined and modified over the intervening 500 million years, but they have never been fundamentally reinvented. The Cambrian was not just the birth of diversity, it was the birth of complexity itself, the moment when life stopped being simple and became extraordinary. But before we dive into those ancient oceans, if you're finding this journey through deep time interesting, consider subscribing to continue exploring the worlds that shaped our own. And let me know in the comments where you're listening from, your city, your time zone. It's always fascinating to see who's joining us from around the world, all connected by curiosity about a time 500 million years ago. Now, to understand how dramatic the Cambrian explosion truly was, we need to see what came before it. We need to travel back even further to the world of the Ediacaran period, a time when life was abundant but bore almost no resemblance to anything we would recognize today. The Ediacaran period, stretching from roughly 635 to 541 million years ago, represented the final chapter of a much longer era when life remained stubbornly simple. The Ediacaran oceans were not empty, far from it, but the creatures that inhabited them would look profoundly strange to modern eyes, so strange that when their fossils were first discovered, scientists struggled for decades to understand what they were looking at. The Ediacaran fauna, as these organisms came to be known, were soft-bodied creatures that lived on or just above the seafloor, and their body plans followed rules that no longer exist in nature. Many of them were flat, almost two-dimensional, with quilted or segmented surfaces that gave them the appearance of air mattresses or geometric patterns pressed into the mud. Some, like Dickinsonia, grew to nearly a meter in length despite having no obvious mouth, no gut, no organs of any kind that we can identify. Others, like Charnia, looked more like ferns or fronds, anchored to the seafloor and extending upward into the water column, though they were animals, not plants. These creatures lived in a world without predators, and this fact shaped everything about them. There was no need for shells, no need for speed, no need for eyes or any sensory apparatus to detect danger, because danger, in the form we understand it today, simply did not exist. The seafloor of the Ediacaran was dominated by microbial mats, those same thick carpets of cyanobacteria that had covered the planet for billions of years, and the Ediacaran animals seem to have lived in harmony with these mats, either feeding on them directly or absorbing nutrients through their flattened bodies. It was a peaceful world, almost pastoral in its simplicity, where life followed predictable rhythms and competition took subtle, passive forms. The absence of hunting, of chasing, of any violent struggle for survival meant that the Ediacaran represented a kind of biological equilibrium, a stable state that might have continued indefinitely, if not for the changes that were quietly building beneath the surface. Those changes were already underway, though they would take millions of years to fully manifest. The oxygen content of the atmosphere and oceans had been rising steadily throughout the late Proterozoic, thanks to the photosynthetic activity of cyanobacteria and the burial of organic carbon in seafloor sediments. This oxygen was accumulating slowly, inexorably, and while it had not yet reached modern levels, it was approaching a critical threshold, a point beyond which entirely new forms of life would become possible. At the same time, the chemistry of the oceans was shifting in subtle but important ways. The concentration of calcium in seawater was increasing, driven by the weathering of continental rocks and the influx of minerals from rivers and hydrothermal vents. This calcium would prove essential for what was coming, because it provided the raw material that animals would soon learn to use in ways that had never been attempted before. Toward the very end of the Ediacaran, around 550 million years ago, the first hints of this coming transformation began to appear in the fossil record. Small, tube-shaped organisms called Cloudina started building external skeletons made of calcium carbonate, simple protective structures that marked the beginning of biomineralization, the process by which living organisms incorporate minerals into their bodies. These early skeletons were crude by later standards, little more than stacked cones or tubes, but they represented a fundamental innovation, a new strategy for survival that would soon be adopted and refined by countless other lineages. At roughly the same time, trace fossils began to appear in the sediments, burrows and tracks left by small worm-like creatures that were beginning to explore the space beneath the microbial mats, tunneling through the mud in search of food or shelter. These burrows were shallow at first, just a few millimeters deep, but they marked the beginning of a process that would eventually revolutionize the structure of marine ecosystems. The Ediacaran world was changing, though the changes were gradual enough that they might not have been obvious to an observer watching in real time. The oxygen levels continued to climb while the calcium concentration in the oceans rose steadily, and as these chemical changes accumulated, the first simple predators began to appear, small creatures that fed on the abundant bacteria and perhaps on each other, introducing the concept of predation into ecosystems that had never known it before. The climate, which had been relatively stable, began to fluctuate as the boundary between the Ediacaran and the Cambrian approached. Sea levels rose and fell. Ocean currents changed their patterns, mixing nutrients from the deep ocean into the shallow coastal waters where most life existed. The microbial mats, which had dominated the seafloor for billions of years, began to break up as the burrowing animals churned the sediments and disrupted their structure. These were the conditions that set the stage for the Cambrian Explosion, a world on the edge of transformation, where the old rules were beginning to break down and new possibilities were opening up. The Ediacaran fauna, those strange quilted creatures that had thrived in the absence of predation, began to decline and eventually disappear, unable to adapt to a world that was becoming more complex, more competitive, and more dangerous. The microbial mats that had provided a stable foundation for life were giving way to a more dynamic, three-dimensional environment where animals could burrow below the surface and swim above it, exploiting new ecological niches that had never existed before. The long, quiet era of simple life was ending, and the planet was about to witness an explosion of innovation unlike anything that had come before or would come after. As we cross that boundary, as we move from the Ediacaran into the earliest moments of the Cambrian, we can almost feel the tension building, the sense that something profound is about to happen. The ingredients are all in place now: the oxygen, the calcium, the genetic toolkit that would allow animals to build complex body plans, the ecological pressures that would drive rapid evolution. The stage is set. The actors are waiting in the wings, and the curtain is about to rise on one of the most extraordinary performances in the history of life on Earth. The evidence for what happened during the Cambrian explosion comes from many sources, from scattered fossils found in rocks around the world, from chemical signatures preserved in ancient sediments, from the genetic relationships between modern animals that allow us to trace their lineages backward through time. But if you want to truly see the Cambrian, if you want to stand face to face with the creatures that emerged during this explosion and understand the world they inhabited, there is one place above all others that you need to visit. High in the Canadian Rockies, in British Columbia's Yoho National Park, there exists a thin layer of rock that has preserved a moment in deep time with such extraordinary clarity that it fundamentally changed our understanding of early animal life. This is the Burgess Shale, and its discovery represents one of the most important events in the history of paleontology. The story of the Burgess Shale begins on a late summer day in August 1909, when an American paleontologist named Charles Doolittle Walcott was riding on horseback through the mountains near the town of Field, British Columbia. Walcott was already a distinguished scientist, the secretary of the Smithsonian Institution, and he had spent years studying Cambrian fossils from various sites around North America. On this particular afternoon, as he and his wife were making their way down a mountain trail, one of their horses allegedly dislodged a block of shale that split open to reveal the fossilized impression of a soft-bodied creature, preserved in remarkable detail. The exact circumstances of this discovery have been embellished over the years, and the story of the horse's hoof may be apocryphal. But what matters is that Walcott recognized immediately that he had found something extraordinary. Over the following summers, from 1910 to 1924, Walcott returned to the site again and again, excavating a quarry high on the slopes of what is now called Fossil Ridge. He worked in conditions that would test anyone's endurance at an elevation of over 2,300 meters, where the weather could turn violent without warning, and the working season lasted only a few months each year. Despite these challenges, Walcott and his team extracted more than 65,000 specimens from the Burgess Shale, carefully wrapping each fossil and shipping them back to Washington, D.C., where they were catalogued and stored in the collections of the Smithsonian. What Walcott had discovered was a fossil site of exceptional preservation, a place where not just the hard shells and skeletons of ancient animals had been preserved, but their soft tissues as well, the muscles and gills and digestive tracts that normally decay within days of death and leave no trace in the geological record. The Burgess Shale formed roughly 508 million years ago during the middle of the Cambrian period, and the conditions that allowed it to preserve soft tissues were unusual and precise. The animals lived in a shallow marine environment near the base of a massive underwater cliff, part of the ancient continental margin where the seafloor dropped away into deeper water. Periodically, perhaps triggered by storms or earthquakes, underwater mudslides would cascade down this cliff, picking up animals from the shallows and carrying them rapidly into the deep, oxygen-poor waters below. In this low-oxygen environment, the bacteria that would normally decompose organic tissue could not survive, and the fine mud that buried the animals protected them from scavengers and mechanical disruption. Over millions of years, as more sediment accumulated on top of these buried organisms, the pressure transformed the mud into shale, and the soft tissues of the animals were replaced molecule by molecule with minerals, creating fossils that preserve details down to the level of individual cells. When you look at a Burgess Shale fossil in a museum case or in photographs, what strikes you immediately is the level of detail. You can see the segmented bodies of arthropods, the feathery gills that would have extracted oxygen from the water, the compound eyes with their individual facets, even the remains of the last meal in the gut of a predator. These are not vague impressions or fragmentary remains. These are complete animals, frozen in time, allowing us to reconstruct not just what they looked like, but how they lived, what they ate, how they moved through the water. The Burgess Shale opened a window into the Cambrian seas that revealed a world far stranger and more diverse than anyone had imagined. Among the tens of thousands of specimens that Walcott collected, certain creatures stood out for their bizarre anatomy and the questions they raised about the nature of early animal evolution. The apex predator of these ancient seas was Anomalocaris, whose name means strange shrimp, though it took decades for scientists to realize that this was a single animal rather than several separate fossils that had been incorrectly identified. Reaching lengths of up to a meter, enormous by the standards of the time, Anomalocaris hunted with a sophistication that surprised researchers when they finally understood its anatomy. It possessed a circular mouth surrounded by plates that could close like an iris, grasping appendages at the front of its body that could catch and manipulate prey, and large compound eyes mounted on stalks, giving it the ability to track movement in the water with remarkable precision. This was not a primitive experiment in predation, but a refined hunter, an animal that required complex sensory systems and coordinated movement, demonstrating that the evolutionary arms race of the Cambrian had already produced highly specialized killing machines. Equally bizarre was Opabinia, a creature so strange that when it was first presented at a scientific conference in 1972, the audience reportedly burst into laughter, unable to believe that such an animal could have existed. With 5 eyes arranged across the top of its head, and a long, flexible proboscis extending from the front of its body, tipped with grasping claws, Opabinia looked like something from a science fiction film, yet the fossils were clear and detailed, leaving no doubt that this was a real animal that had lived and died in the Cambrian seas. Hallucigenia earned its name from its dreamlike, almost hallucinatory appearance, with spines projecting from its back and what appeared to be tentacles or legs extending from its underside, though paleontologists initially reconstructed it upside down, a mistake that persisted for years until better specimens were discovered and the true orientation became clear. The diversity of body plans extended far beyond these celebrated oddities. Wiwaxia, covered in protective scales and spines, resembled something between a mollusk and an annelid worm, defying easy classification and suggesting that the Cambrian seas were home to experimental designs that would eventually be abandoned by evolution. Marella, the lace crab, became the most abundant fossil in the Burgess Shale collections, a small arthropod with elaborate head shields and trailing appendages that filtered food from the water as it swam. Perhaps most significant for our own story was Pikaia, an unassuming creature barely a few centimeters long that swam through the water with undulating movements of its flattened body. Pikaia possessed a notochord, a flexible rod running along its back that provided support for muscles and allowed for more efficient swimming, making it one of the earliest known chordates and placing it within our own lineage, a humble beginning for a group that would eventually give rise to fish, amphibians, reptiles, mammals, and ultimately humans. The Burgess Shale revealed a Cambrian world that was not just diverse in terms of the number of species present, but diverse in terms of the range of body plans and ecological strategies that had evolved. Filter feeders and predators coexisted with burrowers and swimmers, while creatures that crawled along the seafloor shared space with those that drifted in the water column. The ecological complexity of these ancient seas was comparable to modern marine environments, with multiple trophic levels, complex food webs, and a variety of adaptations for capturing food, avoiding predators, and reproducing. This was not a primitive world of simple, experimental organisms fumbling toward complexity. This was a mature ecosystem, fully functional and sophisticated, operating according to the same basic principles that govern life today. Yet the Burgess Shale, remarkable as it was, represented only the middle stages of the Cambrian Explosion. By the time these animals lived and died 508 million years ago, the revolution was already well underway, the major innovations already established. To see the explosion at its earliest moments, to witness the very beginning of this transformation, we need to travel even further back in time and across the world to another extraordinary fossil site, one that predates the Burgess Shale by more than 10 million years and offers an even earlier glimpse into the origins of animal complexity. The Burgess Shale had shown the world what the Cambrian seas looked like in their middle stages, roughly 508 million years ago, when the explosion was already well underway and complex ecosystems had become established. For decades, it stood as the primary window into this ancient world, the most complete record we had of early animal life. But paleontologists knew that the Burgess Shale represented only part of the story, a snapshot taken after many of the crucial innovations had already occurred. To understand the true beginning of the Cambrian explosion, to see the moment when complexity first emerged, scientists needed to find fossils that were older, deposits that captured life as it was transitioning from the simple Ediacaran forms to the sophisticated animals that would dominate the Cambrian seas. That discovery came in 1984 on the other side of the world, in the Yunnan province of southwestern China. A Chinese paleontologist named Hu Xiangguang was examining rock formations near the small town of Chengjiang when he found something remarkable. The rocks he was studying, part of what geologists call the Maotian Shan shales, contained fossils with the same exceptional preservation as the Burgess Shale, soft tissues and delicate structures captured in stunning detail. But these fossils were significantly older, dating to approximately 520 million years ago, pushing the record of complex animal life back by more than 10 million years and placing it much closer to the very beginning of the Cambrian period. What Hu had discovered was not just another fossil site, but a window into the earliest phases of the explosion itself, a time when the innovations that would define animal life were still fresh and experimental. The Chengjiang fossil beds, as they came to be known, have since yielded an extraordinary diversity of specimens, with more than 196 species documented across at least 16 different phyla, representing virtually every major group of animals that existed during the early Cambrian. The preservation at Chengjiang rivals and in some ways surpasses that of the Burgess Shale, with fossils showing not only external anatomy but internal structures as well, including digestive systems, nervous tissue, and even, in some remarkable cases, traces of the animal's brains. Modern imaging techniques, particularly micro-CT scanning, have allowed researchers to examine these fossils in 3 dimensions without damaging them, revealing details that would have been impossible to see even a few decades ago. The result has been a revolution in our understanding of how quickly animal complexity emerged and how sophisticated these early ecosystems actually were. What makes Chengjiang particularly significant is not just its age or the quality of its preservation, but the fact that it contains creatures representing evolutionary experiments that were happening in real time, animals that were trying out different solutions to the challenges of survival in this newly competitive world. Among the most important discoveries at Chengjiang are the early Chordates, animals that possessed the defining features of our own lineage. Haikuichthys and Milokunmingia, both found in the Chengjiang deposits, are among the earliest known fish-like vertebrates, small creatures barely a few centimeters long that swam through the water using muscle blocks arranged along a notochord. These animals had gills for extracting oxygen from the water, sensory organs concentrated at the front of the body, and a basic but functional arrangement of internal systems that would eventually be refined over hundreds of millions of years into the body plan shared by all vertebrates, including humans. The fact that these early chordates appear at Chengjiang, so close to the beginning of the Cambrian, tells us something profound about the speed of the explosion. These were not simple organisms. They had complex anatomy, specialized organs, and sophisticated behaviors, yet they existed only 20 million years after the Cambrian began, a time span that seems impossibly short for evolution to have produced such complexity from the simple Ediacaran ancestors that preceded them. The Chengjiang fauna demonstrates that the Cambrian explosion was not a gradual process that unfolded over tens of millions of years, but a rapid burst of innovation that happened with startling speed, as though life had been waiting for some signal, some trigger that would allow it to finally explore all the possibilities that had been closed off during the long ages when complexity remained stubbornly out of reach. Beyond the chordates, Chengjiang preserves a remarkable diversity of arthropods, the group that would become the most successful and numerous animals in the Cambrian seas. These early arthropods show an astonishing variety of body plans, with different species experimenting with different numbers of segments, different arrangements of appendages, and different strategies for feeding and locomotion. Some were predators, equipped with grasping claws and large eyes. Others were filter feeders, using modified legs covered in bristles to strain microscopic food particles from the water. Still others were scavengers, crawling along the seafloor and feeding on detritus and dead organisms. This diversity suggests that the fundamental ecological roles that would structure marine communities for the rest of Earth's history had already been established by 520 million years ago, with animals occupying every available niche and competing for resources in ways that drove continued evolution and refinement of their adaptations. Perhaps the most extraordinary aspect of the Chengjiang fossils is what they reveal about the evolution of the nervous system and sensory organs. In 2012, researchers examining a fossil of Fuxianhuia pratensa, an arthropod from the Chengjiang deposits, announced that they had found preserved traces of the animal's brain and nerve cords, structures that had been mineralized and fossilized along with the rest of the body. This was the oldest neural tissue ever discovered, and it showed that even at this early stage, Cambrian animals had developed complex nervous systems capable of processing sensory information and coordinating sophisticated behaviors. The presence of large, well-developed eyes in many Chengjiang fossils further emphasizes this point, demonstrating that vision had become a critical sense by the early Cambrian and that animals were using it to navigate their environment, find food, and avoid predators. The Chengjiang site also preserves evidence of complete food webs, with predators, prey, filter feeders, and decomposers all represented in the fossil assemblage. This ecological complexity is what makes the Cambrian explosion so remarkable. Not just that complex animals appeared, but that they immediately organized themselves into functioning ecosystems with multiple trophic levels and intricate relationships between species. The world of Chengjiang was not a transitional phase, not a primitive experiment still finding its footing. It was a fully realized marine ecosystem, operating with the same basic principles that govern ocean life today, and it existed barely 20 million years after the Cambrian began. This suggests that once the trigger for complexity was pulled, once the conditions aligned to allow animals to diversify and specialize, the process unfolded with remarkable speed, filling every available niche and establishing patterns that would persist for hundreds of millions of years. The discoveries at Chengjiang have fundamentally changed our understanding of the Cambrian explosion, pushing its origins back in time and demonstrating that the rapid diversification of animal life was even faster and more dramatic than scientists had previously believed. The fossils from these Chinese deposits show us that by 520 million years ago, the world had already been transformed. The simple, two-dimensional existence of the Ediacaran replaced by a three-dimensional ocean filled with swimming predators, burrowing organisms, and complex interactions between species. The stage was set, the players were in place, and the drama of animal life had begun in earnest. But even with all that the Burgess Shale and Chengjiang have revealed about the Cambrian world, questions remain. The most pressing of these questions concerns not what happened, but how it happened. What innovations made this explosion possible? What evolutionary breakthroughs allowed animals to suddenly develop the features that define complex life? Features like eyes and shells and sophisticated nervous systems. To answer these questions, we need to examine one of the most important innovations of the Cambrian, an adaptation that would change the nature of life on Earth forever and create an arms race that would drive evolution for the next 500 million years. Among all the innovations that emerged during the Cambrian Explosion, few had as profound an impact on the trajectory of life as the evolution of vision. The ability to see, to gather information about the environment through the detection of light, fundamentally transformed the nature of existence for animals in the Cambrian seas. Before eyes, life in the ocean operated largely through touch and chemical sensing, with organisms detecting food or threats only when they came into direct contact or when molecules drifted close enough to trigger a response. This was a world of limited awareness, where danger could approach from any direction without warning, and where finding food required either waiting for it to drift within reach or blindly searching through the water. The evolution of eyes changed everything, creating a new dimension of awareness that allowed animals to detect predators at a distance, to track moving prey, and to navigate complex three-dimensional environments with precision. The fossil record shows us that eyes appeared with startling rapidity during the early Cambrian, and not as simple light-sensitive spots that gradually evolved into more complex structures over vast spans of time, but as sophisticated organs that seem to have been fully functional from the moment they first appear in the rocks. Trilobites, those ubiquitous arthropods that would dominate the Cambrian seas for millions of years, possessed compound eyes from their earliest appearance around 521 million years ago, and these were not primitive prototypes but highly refined optical systems containing hundreds or even thousands of individual lenses, each one a precisely shaped crystal of calcite that focused light onto photoreceptive cells beneath. The sudden appearance of such complex structures troubled Darwin and continues to fascinate evolutionary biologists today, not because it suggests anything supernatural or unexplainable, but because it demonstrates how quickly natural selection can work when the selective pressure is strong enough and the raw materials are available. The trilobite eye represents one of evolution's most elegant solutions to the challenge of vision. Unlike the eyes of most modern arthropods, which use organic lenses made of protein and chitin, trilobites constructed their lenses from calcite, a crystalline form of calcium carbonate that happens to have optical properties suitable for focusing light. Each lens in a trilobite's compound eye was a single crystal, precisely oriented to take advantage of calcite's natural refractive properties, and the array of these lenses covered a curved surface that gave the animal a wide field of view, allowing it to detect movement across a broad arc of its surroundings. The most sophisticated trilobite eyes, found in species that lived during the Ordovician and Devonian periods, contained up to 15,000 individual lenses, each one perfectly shaped and positioned to contribute to the overall image. The optical design of these eyes was so sophisticated that it independently arrived at solutions that human engineers would not discover until the 17th century. Some trilobites developed what is known as a doublet lens structure, where each visual unit contained two lenses with different refractive indices working together to correct for spherical aberration. The distortion that occurs when light passing through the edges of a lens focuses at a different point than light passing through the center. This is the same principle that René Descartes and Christian Huygens described in their work on optics. Yet trilobites were using it 400 million years before humans understood the mathematics behind it. The fact that natural selection could produce such refined optical systems through purely mechanical processes testing random variations and preserving those that provided even slight advantages in detecting food or avoiding predators, demonstrates the power of evolution to generate complexity when the conditions are right. But trilobites were not the only animals experimenting with vision during the Cambrian. The apex predator Anomalocaris, that meter-long hunter we encountered in the Burgess Shale, possessed compound eyes that may have been even more impressive than those of trilobites, with some specimens showing eyes that were large enough to have contained up to 30,000 individual lenses. These eyes were mounted on short stalks that could move independently, giving Anomalocaris the ability to track prey while swimming, a level of sensory sophistication that speaks to the evolutionary arms race that was already well underway by the Middle Cambrian. Fossils of Anomalocaris eyes discovered in South Australia have been preserved in 3 dimensions, allowing researchers to examine their structure in detail and confirming that these were functional organs capable of forming detailed images, not just detecting light and shadow, but actually resolving shapes and tracking movement through the water. The evolution of eyes created what biologists call an evolutionary arms race, a feedback loop where improvements in one group of animals drove corresponding improvements in others. Once some animals could see, prey species faced intense selective pressure to develop their own eyes or to evolve other defenses against visual predators, while predators faced pressure to improve their vision to keep pace with increasingly evasive prey. This dynamic drove rapid innovation in sensory systems, body plans, and behaviors, contributing to the explosive diversification that characterizes the Cambrian. Eyes made hunting more efficient, which meant that predation became a more significant factor in shaping ecosystems, which in turn favored the evolution of shells, spines, burrowing behaviors, and all the other defensive adaptations that appear in the Cambrian fossil record. The impact of vision extended beyond the direct advantages it provided to individual animals. Eyes changed the nature of ecological interactions, creating new possibilities for cooperation, competition, and communication. Animals that could see each other could recognize potential mates, assess rivals, and coordinate group behaviors in ways that had been impossible in the pre-visual world. The ability to perceive color, which some Cambrian animals may have possessed based on the structure of their eyes, would have allowed them to use visual signals for communication, displaying bright patterns to attract mates or warning colors to deter predators. The 3-dimensional ocean that had been largely invisible to its inhabitants suddenly became a visual landscape, filled with information that could be processed and acted upon. The speed with which complex eyes evolved during the Cambrian suggests that the genetic and developmental machinery for building such structures was already present in the common ancestors of Cambrian animals, waiting to be activated and refined. Modern research in evolutionary developmental biology has shown that the same master control genes, particularly a gene called PAX6, are involved in eye development across a wide range of animal groups, from insects to mollusks to vertebrates. This suggests that the basic genetic toolkit for making eyes was inherited from ancient ancestors, and what happened during the Cambrian was not the invention of vision from scratch, but the activation and elaboration of existing developmental programs in response to new selective pressures. Once the conditions in the Cambrian seas favored the evolution of eyes, multiple lineages independently modified their shared genetic inheritance to produce visual organs, each one adapted to their particular needs and lifestyles. The presence of sophisticated eyes in the earliest Cambrian fossils tells us something important about the nature of the explosion. This was not a period of gradual experimentation where simple forms slowly gave rise to complex ones over immense stretches of time. The Cambrian was a period of rapid innovation where complex adaptations appeared quickly, sometimes seemingly overnight in geological terms. because the building blocks were already in place and the selective pressure was intense. Eyes did not evolve slowly through countless intermediate stages. They appeared suddenly because the genes for building them were already present. The environmental conditions favored their development, and the competitive pressure made them essential for survival. Vision became, in a very short time, one of the defining features of animal life, and its influence would shape the evolution of life on Earth for the next 500 million years, driving the development of nervous systems, brains and behaviors of ever-increasing sophistication. The evolution of eyes gave animals the ability to see danger approaching, but vision alone was not enough to survive in a world where predators had become sophisticated hunters. Once animals could track their prey through the water, once they could coordinate attacks and pursue fleeing victims with precision, the selective pressure on potential prey became intense. The soft-bodied organisms that had thrived in the peaceful Ediacaran world suddenly found themselves vulnerable in ways they had never experienced before, and evolution responded with one of the most important innovations in the history of life. Animals began to build armor, protective structures made from minerals extracted from seawater and incorporated into their bodies through the process we now call biomineralization. This was not a single invention that spread from one lineage to others, but rather a solution that evolution discovered independently dozens of times, as different groups of animals arrived at the same basic strategy through different developmental pathways. The fossil record shows us that biomineralization appeared multiple times during the late Ediacaran and early Cambrian, with at least 36 separate origins of mineralized skeletons documented across various animal groups. This remarkable convergence tells us that the raw materials and the selective pressure were both present, waiting for evolution to stumble upon the right combination of genes and developmental processes that would allow organisms to manipulate calcium, silica, or phosphate into protective structures. The timing of these innovations correlates closely with the rise of predation, suggesting that the primary driver was defense rather than structural support or protection from environmental factors. Though once animals possessed hard parts, they could exploit these structures for multiple purposes. The chemistry of biomineralization depends on the availability of certain minerals in seawater, particularly calcium carbonate, which most marine animals use to construct their shells and exoskeletons. During the Cambrian, the concentration of calcium in the oceans was approximately 3 times higher than it had been during the Precambrian, the result of increased weathering of continental rocks and the transport of dissolved minerals into the sea by rivers. This elevated calcium concentration made it energetically feasible for animals to extract the mineral from seawater and deposit it in organized crystalline structures, creating shells that were strong enough to resist the crushing force of predator jaws or the piercing attempts of spines and claws. The process requires a biological scaffold, typically made of organic molecules like proteins or chitin, onto which the mineral crystals can grow in controlled patterns, and different animal groups evolved different variations on this basic theme. Trilobites constructed their exoskeletons from calcite, the same mineral they used for their eyes, creating a hard, protective covering that encased their bodies while still allowing for flexibility through articulated segments. When a trilobite molted, shedding its exoskeleton to grow larger, it left behind a complete fossil that could be preserved in the rock, which is why trilobites are among the most abundant fossils from the Cambrian and subsequent periods. Mollusks, which include modern snails, clams, and squids, developed shells made primarily of aragonite, another form of calcium carbonate with a different crystal structure than calcite, often layered with organic material to create composite structures that were both strong and lightweight. The nacre or mother of pearl that lines the inside of many mollusk shells is an example of this biomineralized composite, with thin layers of aragonite crystals separated by sheets of protein, that allow the structure to absorb impacts without shattering. Brachiopods, which superficially resemble clams but are actually quite different in their internal anatomy and evolutionary relationships, took a different approach by constructing their shells from calcium phosphate rather than calcium carbonate. This choice of material is unusual among marine invertebrates, and it gave brachiopods shells that were more resistant to dissolution in acidic conditions, though at a higher metabolic cost, since phosphate is less abundant in seawater than calcium. Echinoderms, the group that includes modern sea stars, sea urchins, and sea cucumbers, developed yet another variation by building their skeletons from magnesium-rich calcite arranged in a porous three-dimensional structure called stereom, which combines strength with lightweight and allows the skeleton to grow by adding material to existing structures rather than having to molt and rebuild from scratch. The diversity of biomineralization strategies that evolved during the Cambrian reflects the variety of ecological niches that animals were occupying and the different challenges they faced. Animals that lived on the seafloor needed shells that could resist being crushed by predators or torn apart by water currents, while those that swam in the water column needed to balance protection against weight, since heavy shells would make swimming energetically expensive. Some animals developed spines or tubercles on their shells to make themselves harder to swallow, while others relied on smooth, streamlined shells that reduced drag. The evolution of hard parts also enabled new feeding strategies, since mineralized structures could be used as tools for processing food, scraping algae off rocks, crushing other shelled organisms, or filtering particles from the water. The appearance of mineralized skeletons had profound effects that extended beyond the individual organisms that possessed them. Hard parts changed the physical structure of marine environments by creating new substrates that other organisms could colonize. Shells and skeletons accumulated on the seafloor, forming layers of carbonate sediment that would eventually become limestone, one of the most common sedimentary rocks on Earth. Reefs began to form as certain organisms, particularly a group of sponge-like creatures called archaeocyaths, built calcium carbonate structures that grew upward toward the light and created three-dimensional frameworks that other animals could inhabit. These early reefs were fundamentally different from modern coral reefs, both in their biological composition and their structure, but they served similar ecological functions by increasing habitat complexity and providing shelter for a diverse community of organisms. The evolution of biomineralization also had long-term effects on the global carbon cycle, since calcium carbonate shells represent a significant sink for carbon dioxide, removing it from the atmosphere and oceans and sequestering it in sediments for millions of years. As animals died and their shells accumulated on the seafloor, they created vast deposits of limestone that locked away carbon, potentially influencing global climate patterns and ocean chemistry. The relationship between biomineralization, atmospheric carbon dioxide levels, and climate remains an active area of research, with scientists trying to understand how these processes interacted during the Cambrian and whether changes in the carbon cycle might have influenced the pace and pattern of the explosion itself. The hard parts revolution represents one of those moments in the history of life when a new capability opened up entirely new possibilities and fundamentally changed the rules of existence. Before biomineralization, animals were limited in size and vulnerability, unable to grow too large without collapsing under their own weight, and unable to defend themselves effectively against predators. After biomineralization, animals could experiment with new body plans, new ecological roles, and new strategies for survival. The innovation was so successful that it persists to this day, with billions of organisms in modern oceans constructing shells and skeletons using the same basic chemical processes that evolved during the Cambrian. The beaches we walk on, made of sand composed largely of broken shell fragments, The limestone cliffs that tower over coastlines, the chalk used to write on blackboards, all are legacies of that ancient innovation, physical evidence of the moment when animals learned to build armor and changed the world in the process. The innovations that drove the Cambrian Explosion, the eyes and shells and complex body plans, did not emerge in a static environment. The Earth itself was in motion during this period, with continents drifting across the surface of the planet in response to the slow but relentless forces of plate tectonics. To understand the world where the Cambrian animals lived and evolved, we need to step back and look at the geography of the planet 541 million years ago, a world that would be almost unrecognizable if we could somehow visit it. The familiar shapes of the modern continents did not exist. The map of the Cambrian Earth was entirely different, with landmasses scattered across the globe in configurations that had nothing to do with the arrangement we know today. Just before the Cambrian began, the Earth's landmasses had been assembled into a supercontinent called Rodinia, a vast unified mass that had dominated the late Proterozoic. But Rodinia was already breaking apart by the time the Cambrian started, fragmenting into smaller pieces that would drift away from each other and establish the geography that would define the period. This breakup was not a single catastrophic event, but a process that unfolded over millions of years, driven by the movement of magma in the Earth's mantle and the opening of new ocean basins as the crust stretched and thinned. The rifting of Rodinia created extensive coastlines, shallow continental shelves, and epicontinental seas, environments that would prove ideal for the proliferation of marine life. The largest fragment to emerge from Rodinia's breakup was Gondwana, a massive continent that dwarfed all the others, containing roughly 3 times as much landmass as any other continental block during the Cambrian. Gondwana was assembled from pieces that would eventually become South America, Africa, Antarctica, Australia, India, and Arabia, along with smaller fragments that would later join other continents through subsequent collisions. During the early Cambrian, Gondwana straddled a huge range of latitudes, extending from the South Pole northward across the equator, which meant that it encompassed virtually every climate zone from polar to tropical. As the period progressed, Gondwana rotated counterclockwise, changing its orientation relative to the Earth's rotational axis and shifting the distribution of climate zones across its surface. North of Gondwana lay Laurentia, the second largest continent, which consisted of the core of what would eventually become North America, along with Greenland and parts of Scotland. During the Cambrian, Laurentia was positioned astride the equator, giving it a primarily tropical climate and creating ideal conditions for the formation of extensive shallow seas that covered much of its interior. These epicontinental seas, which flooded the continent as sea levels rose during the early Cambrian, provided vast areas of warm, sunlit, nutrient-rich water where marine life could flourish. The fossil record from Laurentia is particularly rich, with numerous sites preserving Cambrian fauna in remarkable detail, testament to the productive ecosystems that developed in these shallow tropical waters. Baltica, which would eventually become Scandinavia, Eastern Europe, and Western Russia, was positioned at mid to high southern latitudes during the early Cambrian, giving it a cooler climate than either Gondwana or Laurentia. As the period progressed, Baltica rotated significantly, turning more than 60 degrees counterclockwise and drifting northward, which brought it into warmer latitudes and changed the environmental conditions that its coastal ecosystems experienced. Siberia, yet another distinct continental block, sat south of the equator during much of the Cambrian, separated from the other continents by wide expanses of open ocean. Smaller continental fragments, including the blocks that would become North China, South China, and various parts of Southeast Asia, were scattered along the margins of Gondwana, creating an archipelago of smaller land masses in the tropical and subtropical zones. The ocean that surrounded these scattered continents was Panthalassa, a vast body of water that covered most of the Earth's surface and dwarfed even the modern Pacific Ocean in extent. Between the continents, smaller ocean basins opened and closed as the plates moved, with the Iapetus Ocean forming between Laurentia and Baltica, widening as these continents drifted apart. The Proto-Tethys Ocean separated Gondwana from the smaller Asian continental blocks, creating seaways that allowed marine organisms to disperse between different regions. The configuration of continents and oceans during the Cambrian created a complex pattern of ocean currents, with warm equatorial waters flowing between the tropical landmasses and cooler currents circulating at higher latitudes. The tectonic activity during the Cambrian was unusually intense, with continental drift rates that were faster than average for Earth's history. This rapid movement had several important consequences for the evolution of life. First, it created extensive coastlines and shallow seas, environments that are particularly productive for marine life because they receive both abundant sunlight and nutrients from terrestrial weathering. Second, it led to increased volcanic activity along the margins of the continents, which released minerals and nutrients into the oceans and may have contributed to the elevated calcium concentrations that made biomineralization feasible. Third, it created geographic barriers and connections that influenced how organisms could disperse, with ocean basins separating populations and allowing them to evolve independently, while land bridges and shallow seaways allowed migration between regions. The fragmentation of Rodinia and the scattered distribution of continents during the Cambrian created distinct biogeographic provinces, regions where different assemblages of organisms evolved in response to local environmental conditions. The fauna of Gondwana was different from that of Laurentia, which was different from that of Baltica, though there were also cosmopolitan species that managed to disperse across ocean barriers and establish populations on multiple continents. This geographic structure may have contributed to the rapid diversification during the Cambrian Explosion by creating opportunities for allopatric speciation, the process by which populations separated by geographic barriers diverge and eventually become distinct species. As continents drifted and ocean currents shifted, previously isolated populations could come back into contact, leading to competition and further evolutionary innovation. The world of the Cambrian was a dynamic place, not just biologically but geologically, with continents in constant motion and ocean basins opening and closing on timescales of millions of years. This restless planetary surface provided the stage on which the drama of the Cambrian explosion played out, creating diverse environments where different innovations could be tested and refined. The shallow tropical seas of Laurentia, the cooler waters around Baltica, the extensive coastlines of Gondwana. Each region presented different challenges and opportunities, and the organisms that evolved in these varied settings would eventually give rise to the incredible diversity of animal life that would spread across the planet in the periods that followed. The scattered continents of the Cambrian world experienced a climate that was fundamentally different from what we know today, not just in its regional patterns, but in its overall character. This was a greenhouse world, a planet without permanent ice caps at the poles, where warmth extended from the equator to the highest latitudes, and seasonal temperature variations were moderated by the vast expanse of ocean that covered most of the Earth's surface. The Cambrian climate was stable in a way that seems almost unnatural when compared to the wild fluctuations that have characterized more recent geological periods, with no evidence of major glaciations or extreme temperature swings during the 55 million years that the period lasted. This stability, combined with the high sea levels that flooded continental margins and created extensive shallow seas, provided ideal conditions for marine life to diversify and spread across the globe. The warmth of the Cambrian was driven primarily by elevated levels of carbon dioxide in the atmosphere, a greenhouse gas that trapped heat and prevented it from radiating back into space. Estimates based on geochemical proxies suggest that atmospheric CO2 concentrations during the Cambrian may have been 10 to 20 times higher than pre-industrial levels, creating a powerful greenhouse effect that kept global temperatures warm, even at high latitudes where the sun's energy arrived at oblique angles. This CO2 came from volcanic outgassing, the release of gases from the Earth's interior through volcanic eruptions and the gradual degassing of magma at mid-ocean ridges. The breakup of rodinia and the high rate of tectonic activity during the Cambrian meant that volcanic activity was more intense than average, pumping additional carbon dioxide into the atmosphere and maintaining the greenhouse conditions. Without polar ice caps, the Cambrian Earth had no large-scale ice albedo feedback, the process by which white ice reflects sunlight back into space and creates a cooling effect that can trigger further ice growth. Instead, the dark oceans absorbed solar energy efficiently, warming the water and distributing that heat around the globe through ocean currents. The absence of ice also meant that sea levels were high, with estimates suggesting that the oceans stood perhaps 50 to 200 meters higher than they do today, depending on the precise timing within the Cambrian. These elevated sea levels flooded the low-lying portions of the continents, creating epicontinental seas that covered vast areas and provided shallow, warm, sunlit waters where photosynthetic organisms could thrive and support complex food webs. The tropical regions during the Cambrian were likely similar in temperature to modern tropical seas, with surface waters reaching 25 to 30 degrees Celsius, though the lack of modern temperature proxies from this period means that estimates carry significant uncertainty. What is clear from the rock record is that carbonate sediments, which form preferentially in warm tropical waters, were abundant along the coastlines of continents positioned at low latitudes, particularly Laurentia. These carbonate platforms extended for hundreds of kilometers, shallow underwater shelves where calcium carbonate precipitated from seawater and accumulated in thick layers that would eventually become limestone. The organisms living in these tropical seas built the shells and skeletons that contributed to these deposits, creating a feedback loop where life both responded to and shaped its environment. The higher latitudes during the Cambrian were cooler than the tropics, as they are today, but the temperature gradient from equator to pole was much gentler than in the modern world. Studies of fossil distributions and sediment types suggest that even at latitudes of 60 degrees or more, temperatures remained temperate rather than freezing, allowing marine organisms to inhabit coastal waters that would be choked with ice during modern winters. Baltica, which spent much of the Cambrian at high southern latitudes, shows evidence of cool water faunas that were nonetheless diverse and productive, adapted to temperatures that were lower than the tropical norm, but still well above freezing. This relatively uniform temperature distribution meant that marine currents were likely weaker than those in the modern ocean, since the driving force for ocean circulation comes primarily from temperature and salinity differences between equatorial and polar waters. The atmospheric composition during the Cambrian was not just different in terms of carbon dioxide, but also in terms of oxygen, which had been rising throughout the late Proterozoic and continued to increase during the early Cambrian. Estimates suggest that by the beginning of the Cambrian, oxygen levels may have reached 15 to 18% of the atmosphere, approaching but not yet matching the modern level of 21%. This oxygen was produced by photosynthetic organisms, primarily cyanobacteria, and later by early algae, and it accumulated in the atmosphere because it was not being consumed as rapidly as it was being produced. The rise in oxygen had multiple effects on the evolution of animal life, most importantly by making aerobic metabolism more efficient and allowing organisms to grow larger and more active than would have been possible in a low-oxygen environment. Seasonal patterns during the Cambrian are difficult to reconstruct with precision, but the position of continents relative to the equator provides some clues. Laurentia, straddling the equator, would have experienced minimal seasonal variation in temperature and day length, creating a stable environment where organisms could reproduce year-round without having to cope with harsh winters or dramatic shifts in food availability. Gondwana, stretching from the South Pole to the tropics, would have experienced a much wider range of seasonal patterns, with its southern regions facing long winter nights, during which photosynthesis would have slowed or stopped entirely, while its equatorial regions enjoyed perpetual warmth. These differences in seasonal patterns may have influenced the types of organisms that could survive in different latitudes and contributed to the biogeographic patterns that we see in the fossil record. Rainfall patterns during the Cambrian are even more uncertain than temperatures, but the high sea levels and extensive shallow seas suggest that evaporation rates were high, which would have loaded the atmosphere with moisture and likely produced abundant rainfall in coastal regions. The lack of land plants during the Cambrian meant that continental interiors were probably arid, with no vegetation to retain moisture or moderate the climate. Rivers would have flowed from these barren highlands to the sea, carrying dissolved minerals and nutrients that would have fertilized the coastal waters and supported high levels of biological productivity. The weathering of rocks on land, accelerated by the high temperatures and rainfall, released calcium, phosphorus, and other elements that were essential for the growth of marine organisms and the construction of shells and skeletons. The stable, warm climate of the Cambrian persisted for most of the period, providing a consistent backdrop against which the drama of the explosion unfolded. This stability was eventually broken near the end of the Cambrian, when evidence suggests a cooling event that may have been associated with changes in ocean circulation or a temporary drawdown of atmospheric carbon dioxide. This end Cambrian cooling, while not a full-scale glaciation, was enough to stress marine ecosystems and contribute to a series of extinction events that marked the transition from the Cambrian to the Ordovician period. But for the vast majority of the Cambrian, the Earth was a greenhouse world, warm from pole to pole, with high sea levels and abundant shallow seas, a planetary environment that proved ideal for the proliferation of complex animal life and the establishment of ecological patterns that would endure for hundreds of millions of years. The warm, stable climate and the scattered geography of the Cambrian world created environments where life could flourish, but it was the organization of that life into functioning ecosystems that truly demonstrated how far evolution had come since the simple days of the Ediacaran. By the time the Burgess Shale and Chengjiang deposits were forming, marine ecosystems had developed a complexity that rivaled modern oceans, with multiple trophic levels, intricate feeding relationships, and a diversity of ecological strategies that covered virtually every possible way to make a living in the sea. This was not a world of random interactions between unconnected species, but a tightly woven network where the success or failure of one group could cascade through the entire system, affecting organisms that might never directly encounter each other. At the foundation of every marine ecosystem lies primary production, the conversion of sunlight and dissolved nutrients into living tissue through photosynthesis. During the Cambrian, this fundamental process was carried out primarily by cyanobacteria and various types of algae, both microscopic single-celled forms drifting in the water column as phytoplankton and larger multicellular forms attached to the seafloor or floating near the surface. These photosynthetic organisms captured energy from the sun and used it to build organic molecules from carbon dioxide and water, creating the biochemical foundation that would support every other organism in the ecosystem. The productivity of Cambrian seas was likely very high in coastal regions, where nutrients washed down from the continents and sunlight penetrated the shallow water, creating conditions similar to the most productive ocean regions today. Feeding on this abundance of primary producers were the filter feeders and grazers, organisms that extracted food from the water or scraped it from surfaces. Sponges, which had evolved during the Precambrian, continued to thrive in the Cambrian seas, using specialized cells to pump water through their porous bodies and strain out bacteria and organic particles. These seemingly simple animals were actually complex filter-feeding systems, capable of processing enormous volumes of water and playing a crucial role in nutrient cycling. The archaeocyaths, those enigmatic reef-building organisms that flourished during the early Cambrian, also operated as filter feeders, creating three-dimensional structures that not only provided habitat for other organisms, but also processed water on a massive scale. Trilobites occupied multiple ecological niches within the Cambrian Food Web, with different species adapted to different feeding strategies. Many trilobites were deposit feeders, crawling along the seafloor and sifting through sediments for organic particles, detritus, and the bacteria that decomposed dead material. Their compound eyes would have helped them navigate the seafloor and perhaps detect predators from above, while their legs, equipped with bristles and spines, would have been modified to help move sediment toward their mouths. Other trilobites appear to have been active hunters or scavengers, with some species possessing robust mandibles capable of crushing the shells of small prey items. The diversity of trilobite body plans suggests that these animals had radiated into many different ecological roles, becoming a dominant presence at multiple levels of the food web. The small shellfish and brachiopods that appeared during the Cambrian established themselves as successful filter feeders, attaching to hard substrates or lying on the seafloor and drawing water across their gills to extract suspended food particles. Brachiopods, in particular, developed an efficient feeding mechanism using a specialized structure called a lophophore, a crown of tentacles covered in cilia that created currents to pull water into the shell and capture food. These animals would become extraordinarily successful in the Paleozoic era, with thousands of species eventually evolving to occupy diverse marine habitats. Their abundance in Cambrian deposits demonstrates that the filter feeding strategy was highly effective in the nutrient-rich coastal waters where most marine life concentrated. Above the filter feeders and grazers were the predators, animals that actively hunted other organisms and whose presence fundamentally shaped the behavior and evolution of potential prey. Anomalocaris represented the apex of this predatory guild during the Middle Cambrian, a creature whose size and sophistication had no precedent in earlier periods. With its grasping appendages capable of catching and manipulating prey, its compound eyes tracking movement through the water, and its circular mouth equipped with plates that could process food, Anomalocaris was a formidable hunter. Stomach contents preserved in some Anomalocaris fossils show fragments of trilobite exoskeletons, confirming that these predators fed on the armored arthropods that crawled across the seafloor. The presence of such specialized predators created selective pressure that drove the evolution of defensive adaptations in prey species, leading to thicker shells, sharper spines, and more effective escape behaviors. Smaller predators occupied niches throughout the water column and on the seafloor, with various arthropods developing specialized appendages for catching small prey, worms that hunted through sediments for buried organisms, and possibly some of the early chordates that used their improved swimming abilities to pursue slower-moving animals. The diversity of predatory strategies during the Cambrian suggests that hunting had become a major evolutionary force, with different lineages experimenting with different approaches to the same basic challenge of catching and consuming other organisms. This created an environment where innovation was rewarded, where any adaptation that provided even a slight advantage in capturing prey or avoiding predators would be strongly favored by natural selection. The Cambrian food webs also included organisms that specialized in decomposition and nutrient recycling, though these are harder to identify in the fossil record, since bacteria and fungi rarely preserve well. Nevertheless, we can infer their presence from the ecological logic of the system, since all the organic material produced by photosynthetic organisms and passed up through the food chain would eventually need to be broken down and returned to the dissolved nutrient pool. Decomposers would have been particularly important in the sediments, where dead organisms accumulated and where oxygen levels might have been low enough to slow down but not completely prevent decomposition. The nutrients released by decomposition would then be available for uptake by the next generation of primary producers, completing the cycle. What makes the Cambrian food webs particularly remarkable is not just their complexity, but how quickly they assembled. The Chengjiang deposits, dating to approximately 520 million years ago, show fully developed ecosystems with multiple trophic levels operating within just 20 million years of the Cambrian boundary. This suggests that once the basic innovations of the Cambrian explosion appeared, once animals had eyes and shells and sophisticated nervous systems, the ecological organization followed rapidly. Species did not slowly experiment with different roles over hundreds of millions of years. They quickly sorted themselves into the niches that were available, establishing patterns of predation, competition, and cooperation that would characterize marine ecosystems for the remainder of Earth's history. The Cambrian seas were not a primitive prototype gradually working toward modern complexity. They were fully functional ecosystems operating according to the same fundamental principles that govern ocean life today, and they demonstrate that the essential architecture of animal ecology was established very early in the history of complex life. Among all the animals that emerged during the Cambrian explosion, none would prove more successful or more enduring than the trilobites. These armored arthropods appeared in the fossil record around 521 million years ago, already diverse and widespread, found in marine deposits from virtually every continent. Their sudden appearance, with no obvious ancestors preserved in earlier rocks, troubled early paleontologists who expected to find transitional forms gradually leading up to such complex organisms. What the fossil record shows instead is a group that burst onto the scene already possessing sophisticated compound eyes, articulated exoskeletons, and specialized appendages, ready to dominate the Cambrian seas and continue that dominance for the next 270 million years. The trilobites would outlast the Cambrian, surviving through the Ordovician, Silurian, Devonian, Carboniferous, and into the Permian, making them one of the longest-lived groups in the history of animal life. The name trilobite refers to their most distinctive feature, the division of their bodies into 3 longitudinal lobes running from front to back. A central axial lobe contained the major organ systems and ran like a ridge down the middle of the animal, flanked on either side by pleural lobes that extended outward and often bore spines or other ornamental structures. This 3-part division was consistent across all trilobite species, though the proportions and details varied enormously. The body was also divided transversely into 3 sections: the cephalon, or head shield, at the front, a segmented thorax in the middle that could contain anywhere from 2 to more than 40 segments depending on the species, and a pygidium or tail shield at the rear. This basic body plan proved extraordinarily flexible, allowing trilobites to adapt to a remarkable range of ecological niches through modifications to the size, shape, and ornamentation of these fundamental parts. The exoskeleton that gave trilobites their characteristic appearance was constructed from calcite, the same crystalline form of calcium carbonate they used to build their eyes. This hard mineral shell provided excellent protection against predators and physical damage, though it came with a significant constraint. Unlike modern arthropods that can grow continuously by molting their flexible chitinous exoskeletons and expanding before the new shell hardens, trilobites had to completely shed their rigid calcite armor periodically and construct an entirely new, larger version. The molting process left the trilobite temporarily vulnerable, and the discarded exoskeletons accumulated on the sea floor, which is why trilobite fossils are so abundant. Each individual trilobite might molt dozens of times during its life, and each molted exoskeleton had the potential to be preserved, creating a fossil record far richer than the actual population size would suggest. The diversity of trilobite forms that evolved during the Cambrian reflects the variety of lifestyles they adopted. The earliest trilobites, belonging to groups like the Olenolids that dominated in Laurentia, tended to have large semicircular head shields and relatively few thoracic segments, suggesting they were adapted for crawling along the seafloor in shallow tropical waters. The redlechids, which were more common in the southern hemisphere continents like Gondwana, developed longer bodies with more segments and often possessed prominent spines extending from their pleural lobes, possibly as defense against predators. The agnostids, a peculiar group of very small trilobites, often just a few millimeters in length, had only 2 or 3 thoracic segments and a body plan so simplified that some paleontologists have questioned whether they were actually trilobites at all, though molecular studies of their living arthropod relatives support their placement within the group. Trilobite eyes deserve special attention because they represent one of the most sophisticated visual systems ever evolved. As we discussed earlier, these compound eyes were constructed from calcite crystals, with each lens precisely shaped and oriented to focus light onto the photoreceptive cells beneath. The number of lenses varied dramatically between species, from just a few dozen in some groups to more than 15,000 in the most visually sophisticated forms. The placement of the eyes also varied, with most trilobites having eyes positioned on the dorsal surface of the head shield, giving them an upward and sideways view of their surroundings, though some bottom-dwelling species reduced or lost their eyes entirely, presumably because vision was less useful in the dark sediments where they lived. The evolutionary investment in such complex visual systems tells us that trilobites lived in a visually complex world where the ability to see predators, prey, and potential mates provided significant survival advantages. The appendages of trilobites, which are rarely preserved because they lacked mineralized exoskeletons, appear to have been remarkably versatile structures. Based on exceptional specimens from deposits like the Burgess Shale where soft tissues were preserved, we know that trilobites possessed biramous limbs, appendages with 2 branches, one adapted for walking or swimming and the other modified into a gill for respiration. These limbs lined the underside of the thorax and were probably capable of performing multiple functions, helping the animal move across the seafloor, creating water currents to bring food particles toward the mouth and exchanging gases with the surrounding water. Some trilobites developed specialized anterior appendages near the mouth that may have been used for handling food or for sensory purposes, extending the animal's awareness beyond what the eyes could detect. The geographic distribution of trilobites during the Cambrian reveals patterns that tell us about both ocean currents and continental positions. Certain trilobite groups were cosmopolitan, found on multiple continents, suggesting they had planktonic larval stages that could disperse across ocean basins before settling to the seafloor as adults. Other groups were endemic to specific regions, indicating that geographic barriers prevented their spread, or that they were adapted to environmental conditions that existed only in certain areas. The Olenolids, for instance, were restricted primarily to Laurentia, while the redlechids dominated in Gondwana and the Asian continental blocks. These biogeographic patterns have been used by geologists to reconstruct the positions of continents during the Cambrian and to understand how ocean currents flowed between these land masses. Trilobites played multiple roles in Cambrian ecosystems, occupying positions at several different trophic levels. Many species were deposit feeders, processing sediments to extract organic particles and microorganisms, essentially functioning as underwater earthworms that churned the seafloor and influenced sediment structure. Others were filter feeders that used their limbs to create currents and capture suspended food particles from the water column. Some trilobites were probably scavengers, feeding on dead organisms that settled to the seafloor, while a few species may have been active predators, using strengthened mouthparts to crush or tear apart small prey. The evolution of enrollment, the ability to curl up into a ball by flexing the thorax and bringing the head and tail shields together, provided an effective defense mechanism against predators and is preserved in many trilobite fossils, frozen in their protective posture. The success of trilobites during the Cambrian established them as the dominant arthropods in marine ecosystems, a position they would maintain through most of the Paleozoic era. Their mineralized exoskeletons made them excellent fossils, which means we have an unusually complete record of their evolution and diversification. They provide some of the clearest examples of evolutionary processes preserved in stone, showing gradual changes in body proportions, the appearance and disappearance of spines and ornaments, and adaptations to different environmental conditions. The trilobites that swam and crawled through the Cambrian seas were not primitive experiments in arthropod design, but sophisticated animals with complex anatomy, refined sensory systems, and diverse ecological roles, representatives of a body plan that would prove viable for hundreds of millions of years and that established arthropods as one of the most successful groups in the history of life on Earth. We have traced the evidence of the Cambrian explosion through the fossil beds of Burgess Shale and Chengjiang, examined the innovations that emerged during this period, from eyes to shells to complex nervous systems, and explored the world where these ancient animals lived. But the most fundamental question remains unanswered, a question that has occupied scientists since Darwin first recognized it as a problem for his theory. Why did the Cambrian explosion happen when it did? And why did it unfold with such extraordinary speed? For 3 billion years, life remained stubbornly simple, confined to single cells, and eventually to simple multicellular forms that lacked the complexity we associate with animals. And then, in what amounts to a geological instant, roughly 20 million years, virtually every major animal body plan appeared in the fossil record. This timing demands an explanation, and while scientists have proposed numerous theories over the past century and a half, the emerging consensus is that no single factor can account for the explosion. Instead, it appears to have been the result of multiple causes converging at the same moment in Earth's history, creating conditions that had never existed before and would never be quite the same again. The oxygen hypothesis has been one of the most enduring explanations for the Cambrian explosion, and recent research has both refined and complicated our understanding of its role. Oxygen is essential for complex animal life because aerobic metabolism, the process of extracting energy from food using oxygen, is far more efficient than anaerobic alternatives. A sponge or a worm can survive on relatively low oxygen levels, moving slowly and maintaining a simple body plan that allows oxygen to diffuse through tissues. But an active predator like Anomalocaris, with powerful muscles for swimming, complex eyes that require constant energy to maintain, and a nervous system coordinating rapid movements, needs abundant oxygen to fuel these energy-intensive systems. For most of Earth's history, oxygen levels in the atmosphere and oceans were too low to support such active, complex organisms, remaining below 1% of atmospheric composition for billions of years, while photosynthetic bacteria slowly released oxygen as a waste product of their metabolism. The critical change came during the late Proterozoic, when oxygen levels began to rise more rapidly, though they still had not reached modern concentrations by the time the Cambrian began. Recent estimates suggest that atmospheric oxygen may have reached 15 to 18% by the early Cambrian, compared to today's 21%. While this might seem like a small difference, it represents a threshold beyond which aerobic metabolism becomes efficient enough to support larger, more active bodies. Oxygen also plays a crucial role in collagen synthesis, the process by which animals build the structural proteins that hold tissues together and provide support for complex body plans. Without adequate oxygen, collagen cannot form properly, which means that even if the genetic instructions for building a complex animal were present, the organism would lack the biochemical machinery to actually construct the necessary structures. The rise in oxygen during the late Proterozoic may have finally crossed the threshold where complex animal bodies became physiologically possible. Yet oxygen alone cannot explain the explosion, because oxygen levels had been rising gradually for millions of years before the Cambrian began, and there is no evidence of a sudden spike in atmospheric oxygen coinciding with the appearance of complex animals. This suggests that while oxygen was necessary for the Cambrian explosion, it was not sufficient by itself to trigger it. Other factors had to be involved, working in concert with the oxygen increase to create the conditions for rapid diversification. One of these factors was almost certainly the chemistry of the oceans, particularly the concentration of calcium and other minerals that animals would use to build their shells and skeletons. The weathering of continental rocks during the late Proterozoic released enormous quantities of calcium into the oceans, increasing its concentration to roughly 3 times the levels that had prevailed earlier. This made biomineralization energetically feasible for the first time, allowing animals to extract calcium from seawater and deposit it in crystalline structures without expending prohibitive amounts of energy. The increased availability of calcium created opportunities for innovation that had not existed before. Once some animals began building shells as protection against predators, an evolutionary arms race began, with prey species developing thicker armor while predators evolved stronger jaws and more sophisticated hunting strategies. This feedback loop, known as escalation theory, proposes that predation pressure drove much of the innovation during the Cambrian, with each new defensive adaptation favoring corresponding improvements in offensive capabilities. The fossil record supports this interpretation, showing evidence of predation in the form of healed injuries on trilobite exoskeletons, holes drilled through shells, and the stomach contents of predators containing fragments of armored prey. The appearance of eyes fits naturally into this framework as well, since vision would have been enormously advantageous both for predators tracking their prey and for prey detecting predators at a distance. Another factor that may have contributed to the timing of the Cambrian explosion was the disruption of the microbial mats that had dominated seafloor ecosystems for billions of years. These thick carpets of cyanobacteria and other microorganisms created a two-dimensional living surface that most Ediacaran organisms lived on top of, feeding on the mat itself or absorbing nutrients through their flattened bodies. The evolution of burrowing behavior in the latest Ediacaran and early Cambrian broke up these mats, churning the sediments and creating a three-dimensional environment where organisms could explore space both above and below the seafloor. This Cambrian substrate revolution, as it has been called, fundamentally changed the structure of marine ecosystems and may have opened up new ecological niches that had not previously existed. Animals that could burrow gained access to food resources buried in the sediment, while the disruption of the mats may have reduced competition from the microbial communities and freed up nutrients for animal consumption. The genetic and developmental mechanisms that animals inherited from their ancestors also played a crucial role in enabling the rapid diversification of the Cambrian. Modern research in evolutionary developmental biology, often called Evo-Devo, has revealed that the genetic toolkit for building complex bodies was largely in place before the Cambrian began. The Hox genes, which control the development of body segments and the positioning of limbs and organs along the body axis, are found in nearly identical forms across a wide range of animal groups, from worms to insects to vertebrates. This suggests that these master control genes evolved in the common ancestor of all animals and were inherited by the various lineages that diversified during the Cambrian. What changed during the explosion was not the invention of these genes, but their deployment in new combinations and contexts, creating novel body plans through modifications to the timing and location of gene expression rather than through the evolution of entirely new genetic machinery. This genetic flexibility meant that once environmental conditions favored the evolution of complex bodies, the necessary developmental tools were already available, waiting to be activated and modified. Small changes in the regulation of existing genes could produce dramatic changes in body form, allowing rapid evolutionary innovation without requiring the slow accumulation of entirely new genetic information. The Cambrian Explosion was not a period when the genetic instructions for building animals were written from scratch, but rather a time when existing instructions were edited, remixed, and deployed in creative new ways. The speed of the explosion becomes more comprehensible when we recognize that evolution was not starting from zero, but was instead working with a sophisticated genetic toolkit that had been refined over hundreds of millions of years of earlier evolution. The convergence of these multiple factors, rising oxygen levels, increased calcium availability, the disruption of microbial mats, the pressure of predation, and the activation of existing developmental genes created a perfect storm of conditions that had never occurred before in Earth's history. Rising oxygen enabled the aerobic metabolism required for active, complex bodies, while elevated calcium concentrations made biomineralization energetically feasible for the first time. Predation pressure drove an evolutionary arms race that favored continuous innovation in both offensive and defensive adaptations, and the genetic toolkit inherited from ancient ancestors provided the developmental machinery to rapidly generate new body plans through modifications to existing genes rather than the invention of entirely new ones. The disruption of microbial mats opened up three-dimensional living space and ecological opportunities that had not existed in the stable, mat-dominated world of the Ediacaran. When all of these elements aligned during a relatively brief window in the early Cambrian, life responded with an explosion of creativity and diversity that would establish the basic patterns of animal life for the next 500 million years. The Cambrian seas were populated not only by the ancestors of modern animal groups, but also by creatures so bizarre that when their fossils were first discovered, scientists struggled to place them within any known category of life. These were the organisms that paleontologist Stephen Jay Gould famously called weird wonders in his 1989 book Wonderful Life. Animals whose body plans seemed to represent evolutionary experiments that had no modern descendants, designs that were tried once during the Cambrian and then abandoned, leaving no trace in the subsequent fossil record. Gould argued that these extinct forms demonstrated that the Cambrian was a time of maximum morphological diversity, when evolution was exploring a wider range of body plan possibilities than at any time since, and that much of this early experimentation ended in extinction, leaving us with a modern fauna that represents only a subset of what evolution had originally produced. The most famous of these weird wonders was hallucigenia, an organism so strange that paleontologists initially reconstructed it upside down, mistaking the spines on its back for walking legs and the actual legs for some kind of defensive or sensory structure projecting upward. The creature was barely a few centimeters long, with a worm-like body supported by 7 pairs of legs, each ending in tiny claws, while 7 long spines projected from its dorsal surface, presumably as protection against predators. When the correct orientation was finally established through the discovery of better preserved specimens, hallucigenia still remained deeply puzzling, with no obvious living relatives and no clear place in the evolutionary tree of animals. Its name, derived from the word hallucination, captured the dreamlike quality of its appearance, as though it belonged more to a surrealist painting than to the actual history of life on Earth. Upper Binia presented an equally perplexing puzzle when it was formally described in the 1970s. When paleontologist Harry Whittington showed his reconstruction of the animal at a scientific conference in 1972, the audience reportedly burst into spontaneous laughter, unable to believe that such a creature could have actually existed. Opabinia had 5 eyes arranged across the top of its head, a configuration found in no other known animal, and a long flexible proboscis extending forward from its face, tipped with grasping spines that appeared to function as a feeding appendage. The body was segmented, with lateral lobes extending from each segment that may have been used for swimming, giving the animal a strange undulating appearance as it moved through the water. Despite intensive study, Opabinia's evolutionary relationships remain uncertain, with different analyses placing it near arthropods, near various worm groups, or in a category entirely its own. Wiwaxia added another layer of strangeness to the Cambrian fauna. This small creature, typically just a few centimeters in length, was covered with overlapping scale-like structures called sclerites, arranged in rows across its back, while 2 rows of longer spines projected upward, creating a formidable defensive array. The underside of the animal bore a muscular foot similar to that of modern mollusks, suggesting it crawled slowly across the seafloor, grazing on microbial mats or organic detritus. The combination of mollusk-like features with the spiny armor has made Wiwaxia difficult to classify, with some researchers arguing it represents an early mollusk lineage, while others place it closer to annelid worms. Its exact position in the tree of life remains debated, testament to how difficult it can be to interpret ancient body plans when they do not fit neatly into modern categories. The debate over these weird wonders and their significance has evolved considerably since Gould first brought them to public attention. When Gould wrote Wonderful Life, the prevailing interpretation was that many Burgess Shale organisms represented entirely separate phyla, fundamental body plans that had no connection to any modern animal groups. This view suggested that the Cambrian had produced far more morphological diversity than exists today, and that the history of life involved a progressive narrowing of possibilities, a pruning of the tree that eliminated most of the early experimental forms. However, as more specimens were discovered and analytical techniques improved, many of these supposedly unique organisms were gradually reclassified as early members of known phyla, their strange appearances resulting from unfamiliar combinations of features rather than from truly novel body plans. Hallucigenia, for instance, is now generally accepted as an early relative of the velvet worms, a group of soft-bodied terrestrial animals that still exist today, though in far fewer species than during the Cambrian. The spines and body structure that seemed so alien in the Burgess Shale specimens find parallels in modern velvet worms and in other fossils from the early Paleozoic. Anomalocaris, which had initially been misidentified as 3 separate organisms, a jellyfish mouth, a shrimp body, and mysterious circular structures, is now understood to be a stem arthropod, an early relative of the group that would give rise to modern insects, crustaceans, and their kin. Even Opabinia, despite its 5 eyes and peculiar proboscis, appears to be related to arthropods based on various anatomical features, though its exact position within that radiation remains uncertain. This reinterpretation does not mean that the Cambrian was unremarkable, or that its fauna was simply a primitive version of what exists today. The diversity of the Cambrian was genuinely extraordinary, and many of the body plans that evolved during this period were indeed evolutionary dead ends that left no descendants. What has changed is our understanding of how evolution works and how we should interpret morphological diversity. Rather than representing entirely separate phyla that were later eliminated, many weird wonders appear to be early experiments within existing phyla, stem group members that possessed unusual combinations of features before the more familiar characteristics of each group became fixed. The Cambrian was a time when the basic body plans of modern phyla were still being refined, when different lineages were trying out various solutions to similar problems, and when the boundaries between groups were less distinct than they would later become. The extinction of these strange forms tells us something important about how evolution operates. Not every innovation succeeds in the long term, and not every body plan that functions adequately in one set of environmental conditions will prove viable when conditions change. The organisms that survived the Cambrian and went on to found the major animal lineages we see today were not necessarily superior in any absolute sense. They were simply the ones whose particular combination of features happened to work well enough in the changing environments of the early Paleozoic to avoid extinction and to diversify into new forms. There is an element of contingency in evolution, a role for historical accident and luck, that means the world we inhabit today is not the only possible outcome of the Cambrian explosion, but simply the one that happened to occur. Among all the strange creatures of the Cambrian, perhaps the most significant for our own story were not the ones that seemed most bizarre, but rather the ones that seemed most unremarkable. Small swimming animals barely visible in the fossil record that possessed a simple, flexible rod running down their backs. These were the early chordates, the ancestors of fish, amphibians, reptiles, mammals, and ultimately humans, and their story deserves special attention as we trace the legacy of the Cambrian forward through time. Among the thousands of species that populated the Cambrian seas, most would leave descendants that evolved into the familiar invertebrate groups we see today. The insects and crustaceans descended from Cambrian arthropods, the snails and clams that trace their lineage to early mollusks, the sea stars and sea urchins that emerged from Cambrian echinoderms. But there was one group of organisms easily overlooked among the more numerous and more visually striking creatures of the period that would eventually give rise to a lineage of extraordinary importance. These were the chordates, animals distinguished by a flexible rod called a notochord running along their backs, a dorsal nerve cord, pharyngeal slits in the throat region, and a muscular postanal tail. These features, which seem unremarkable compared to the compound eyes of trilobites or the grasping claws of Anomalocaris, would prove to be the foundation for one of evolution's most successful experiments, the vertebrate body plan that would eventually produce fish, amphibians, reptiles, birds, mammals, and ultimately the species writing and reading these words. The earliest known chordates from the Cambrian appear in the Chengjiang deposits of China, those remarkably preserved fossils dating to approximately 520 million years ago that we examined earlier. Among these ancient organisms are Haikuichthys and Milocunmingia, small fish-like creatures barely 2 to 3 centimeters in length that possessed the defining features of chordates, along with characteristics that would become hallmarks of vertebrates. These animals had segmented muscle blocks called myomeres arranged along either side of a notochord, allowing them to generate the undulating movements necessary for swimming. They possessed pharyngeal slits that may have functioned both for filter feeding and for respiration, extracting oxygen from water as it passed through these openings. And crucially, they showed evidence of a more complex head region than their invertebrate contemporaries, with concentrations of sensory organs and possibly the beginnings of a cartilaginous skull that would protect the enlarged nerve cord at the front of the body. What makes these early Cambrian chordates so significant is not their size or their abundance, since they were vastly outnumbered by arthropods and other invertebrate groups, but rather what they represent in terms of evolutionary potential. The notochord, that simple, flexible rod of stiffened tissue, provided an internal skeleton against which muscles could pull, creating a much more efficient swimming mechanism than was possible for animals that relied on hydrostatic skeletons maintained by fluid pressure. This innovation allowed chordates to become more active swimmers than most of their contemporaries, though they were still far from being the dominant predators of the Cambrian seas. The dorsal nerve cord, positioned along the back rather than along the belly as in most invertebrates, was protected by the notochord and the muscles surrounding it, and its anterior end was already showing signs of enlargement that would eventually lead to the evolution of a brain. The Burgess Shale, dating to approximately 508 million years ago, preserves another crucial piece of the chordate story in the form of Pichia, an organism that Charles Darwin might have appreciated enormously had he known of its existence. Pichia was small, reaching perhaps 4 centimeters in length, with a flattened, blade-like body that undulated as it swam through the water column. The notochord is clearly visible in well-preserved specimens, running the length of the body and providing the structural support necessary for the animal's swimming movements. While Pichia lacked the more advanced features seen in Hyquithis, such as clear evidence of pharyngeal slits or a complex head region, it demonstrates that chordates had achieved a wide geographic distribution by the Middle Cambrian, appearing in both the Asian deposits of Chengjiang and the North American deposits of the Burgess Shale. When we look at the reconstructions of these small swimming animals preserved in the fossil beds of China and Canada, we are looking at organisms that shared with us a common ancestor far back in the mists of deep time, and we are seeing the earliest known expression of the anatomical features that would eventually be elaborated into the human body. The notochord that ran down the back of Hyquithis became the evolutionary precursor of your spine, while the dorsal nerve cord gave rise to your spinal cord and brain, and the pharyngeal slits, though no longer present in their original form, contributed developmental structures to your jaw and ears through a complex series of modifications that unfolded over hundreds of millions of years. Stephen Jay Gould, in his analysis of the Burgess Shale fauna, made much of this contingency. He pointed out that if Pichia had gone extinct during one of the many environmental perturbations that affected marine life during the Cambrian, the entire subsequent history of vertebrate evolution might never have occurred. There would be no fish, no amphibians colonizing the land, no dinosaurs, no mammals, no primates, and no humans to contemplate the history of life on Earth. The fact that we exist at all, Gould argued, is the result of a long chain of historical accidents, beginning with the survival of a small, seemingly insignificant swimming animal in the Cambrian seas. While this view is somewhat oversimplified, since there were multiple chordate lineages in the Cambrian, and the extinction of any single one might not have eliminated the group entirely, it captures an important truth about evolution. Success is not always predictable from initial conditions, and the organisms that seem most impressive or well adapted at any given moment are not necessarily the ones that will leave the most enduring legacy. The transition from these early Cambrian chordates to true vertebrates with mineralized skeletons would take millions of years, unfolding during the Ordovician and Silurian periods as animals began to deposit calcium phosphate in cartilaginous structures, creating bones and teeth. The notochord would eventually be replaced by a segmented vertebral column in most lineages, providing even greater support and flexibility. The pharyngeal slits would be modified into gills in fish, then partially transformed into components of the jaw and ear in terrestrial vertebrates. The dorsal nerve cord would enlarge dramatically at its anterior end, developing into the complex brain that would eventually allow vertebrates to process vast amounts of sensory information and coordinate sophisticated behaviors. But all of these later innovations built upon the foundation established in the Cambrian, the basic chordate body plan that emerged during the explosion and proved versatile enough to be modified and elaborated over the next 500 million years. The early chordates also illustrate a broader pattern in the Cambrian explosion. The fact that many of the major innovations that would define animal life appeared not in large, dominant organisms, but in small, relatively inconspicuous ones. The most impressive animals of the Cambrian, the heavily armored trilobites and the predatory anomalocarids, represent evolutionary paths that would eventually lead to dead ends or diminished importance, while some of the smallest and least conspicuous organisms laid the groundwork for future evolutionary radiations. This reminds us that evolutionary success operates on multiple timescales, with different criteria for success operating in the short term versus the long term. Dominance in the immediate environment does not necessarily translate to long-term evolutionary persistence or diversification. The Chordates of the Cambrian were our ancestors, not in the sense that any individual Pikaia or Hyquicthis was a direct forebear of modern humans, since these specific lineages likely went extinct, but in the sense that they were members of the Chordate clade, possessing the synapomorphies that define our group and representing early experiments with the body plan that would eventually produce the vertebrate radiation. When we look at the reconstructions of these small swimming animals preserved in the fossil beds of China and Canada, we are looking at organisms that shared with us a common ancestor far back in the mists of deep time, and we are seeing the earliest known expression of the anatomical features that would eventually be elaborated into the human body. The notochord that ran down the back of Hyquicthis is the evolutionary precursor of your spine. The dorsal nerve cord is the ancestor of your spinal cord and brain. The pharyngeal slits, though you no longer possess them in their original form, contributed developmental structures to your jaw and ears. The Cambrian did not just establish the basic patterns of marine ecology. It established the anatomical blueprint that would eventually allow life to colonize the land, fill the skies, and develop the cognitive capacity to understand its own evolutionary history. The Cambrian period, which had witnessed such extraordinary innovation and diversification, did not end with a sudden catastrophe. There was no asteroid impact, no massive volcanic eruption, no dramatic event that brought the curtain down on this remarkable chapter of Earth's history. Instead, the Cambrian faded gradually into the Ordovician period around 485.4 million years ago, marked by subtle changes in ocean chemistry, sea level fluctuations, and shifts in the dominant groups of marine organisms. The transition was not smooth, however, and the late Cambrian was punctuated by several extinction events that significantly reduced marine biodiversity and reshuffled the ecological deck, eliminating some of the groups that had dominated the early and middle Cambrian, while creating opportunities for others to diversify and expand. The first major crisis came near the end of the early Cambrian, approximately 509 million years ago, when many of the trilobite groups that had been successful during the initial radiation began to decline. The Olenellids, those distinctive trilobites that had dominated the shallow tropical seas of Laurentia with their large semicircular head shields and elaborate spines, disappeared entirely from the fossil record. The red licheids, which had been equally successful in the southern hemisphere continents, suffered severe losses, though some lineages managed to persist into the Middle Cambrian before finally going extinct. The small shelly fauna that had appeared at the very beginning of the Cambrian, those tiny mollusks and other organisms with simple mineralized structures, also declined dramatically during this interval, suggesting that the environmental conditions that had favored their initial success had changed. The causes of this early Cambrian extinction remain somewhat unclear, though several factors may have contributed. Ocean chemistry was still fluctuating during this period, as the balance between oxygen production by photosynthetic organisms and oxygen consumption by respiration and decomposition continued to shift. Episodes of ocean anoxia, where oxygen levels in deeper waters dropped to levels that could not support most animal life, have been documented from late early Cambrian sediments, and these low oxygen events would have stressed marine ecosystems and potentially triggered extinctions, particularly among organisms that required well-oxygenated water. Changes in sea level may also have played a role, with regressions that drained shallow epicontinental seas, reducing the total area of productive shallow marine habitat and forcing organisms into more crowded and competitive environments. The Middle Cambrian, the time of the Burgess Shale, and the peak diversity of many Cambrian groups, was relatively stable compared to the periods before and after it. This interval, lasting from approximately 509 to 497 million years ago, saw the continued success of trilobites, which diversified into new forms and occupied new ecological niches, while also witnessing the expansion of other arthropod groups, the proliferation of brachiopods and mollusks, and the early radiation of echinoderms. The reef-building archaeocyaths, however, which had been so important during the early Cambrian, declined during the Middle Cambrian and went extinct before the period ended, their ecological role remaining unfilled until new reef-building organisms evolved in later periods. The late Cambrian, beginning around 497 million years ago, brought new challenges for marine life. A series of extinction pulses affected different groups at different times, creating a pattern of gradual biodiversity loss rather than a single catastrophic event. Trilobites, which had been the dominant arthropods throughout most of the Cambrian, experienced significant turnover, with many genera going extinct while new forms evolved to replace them. The extinction was particularly severe for certain ecological specialists, organisms that had adapted to very specific environmental conditions and could not cope when those conditions changed. Bottom-dwelling trilobites that fed on organic particles in the sediment, for instance, may have been affected by changes in ocean circulation that altered the distribution of nutrients and organic matter on the seafloor. The end of the Cambrian, around 485.4 million years ago, was marked by a more significant extinction event that affected a broad range of marine organisms. This end Cambrian extinction, sometimes called the Cambrian Ordovician boundary event, eliminated approximately 40 to 50% of marine genera, though the exact percentage varies depending on which groups are examined and how completeness of the fossil record is assessed. Trilobites again suffered losses, with several major families disappearing, though the group as a whole survived and would go on to diversify again during the Ordovician. Conodonts, small eel-like animals with mineralized tooth-like structures that are abundant in Cambrian and later rocks, showed significant turnover at this boundary. Brachiopods and other filter-feeding organisms experienced extinctions, suggesting that changes in ocean productivity or water chemistry may have been involved. The causes of the end Cambrian extinction are debated, with different researchers emphasizing different factors. Some evidence points to ocean anoxia as a major contributor, with geochemical signatures in rocks from this interval indicating that deep ocean waters became depleted in oxygen, creating dead zones that expanded upward into shallower depths. This oxygen depletion may have been triggered by changes in ocean circulation patterns, possibly linked to the movement of continents and the opening or closing of ocean gateways that affected how water flowed between different ocean basins. Changes in sea level also occurred around this time, with evidence of both regressions that exposed continental shelves and transgressions that flooded them again, creating environmental instability that would have stressed marine ecosystems. Climate change may have played a role as well, though the evidence is less clear than for some later mass extinctions. The Cambrian had been characterized by a warm, stable greenhouse climate with no polar ice, but some studies suggest that cooling began in the late Cambrian, perhaps driven by changes in atmospheric carbon dioxide levels or by shifts in ocean heat transport. This cooling was not dramatic enough to trigger a full glaciation as would occur in the Ordovician, but even modest temperature changes can affect marine ecosystems by altering the distribution of nutrients, changing the timing of seasonal productivity cycles, and shifting the geographic ranges where different organisms can survive. Despite these extinction events, the Cambrian should not be viewed as a failure or as a period whose innovations were largely wiped out. The groups that survived the late Cambrian extinctions went on to dominate the early Paleozoic oceans, diversifying and evolving new forms that would prove even more successful than their Cambrian ancestors. Trilobites, though they lost diversity at the end of the Cambrian, rebounded during the Ordovician and reached their peak diversity in terms of total number of species. Brachiopods expanded dramatically, becoming the most abundant shell-bearing organisms in many marine environments. Echinoderms, which had been relatively minor players in the Cambrian, diversified into the many forms that would characterize them throughout the Paleozoic. And the chordates, those small swimming animals that seemed so insignificant during the Cambrian, began their long evolutionary journey toward the vertebrate radiation that would eventually produce fish, then tetrapods, then mammals. The end of the Cambrian was not an ending but a transition, a passage from one phase of animal evolution to another. The innovations that appeared during the Cambrian, eyes and shells and complex nervous systems and sophisticated body plans, persisted and continued to evolve. The ecological patterns established during the Cambrian, the food webs with multiple trophic levels, the arms races between predators and prey, the partitioning of environments into different niches occupied by specialists, all of these became permanent features of life on Earth. The Cambrian explosion had created the basic architecture of animal life, and what followed in subsequent periods was not a replacement of that architecture, but an elaboration and refinement, a working out of the possibilities inherent in the body plans and ecological relationships that first emerged during those remarkable 55 million years when life decided to become complex. While the Cambrian explosion produced countless innovations and experiments in body plan design, and while many of these experiments ended in extinction or left only modest legacies, one group of organisms established a dynasty that would endure for an almost incomprehensible span of time. The trilobites, those armored arthropods that appeared suddenly in the fossil record around 521 million years ago, would continue to inhabit the world's oceans for the next 270 million years, surviving through the Ordovician, Silurian, Devonian, Carboniferous, and well into the Permian period before finally succumbing to the greatest mass extinction in Earth's history. To put this longevity in perspective, trilobites thrived for a period approximately 54 times longer than the entire existence of our own genus Homo, and they witnessed the rise and fall of countless other groups while adapting, evolving, and maintaining their basic body plan through environmental changes that would have eliminated less versatile organisms. The success of trilobites during the Ordovician period, which followed the Cambrian, demonstrates how the innovations of the Cambrian explosion could be refined and elaborated over geological time. The Ordovician, lasting from 485.4 to 443.8 million years ago, saw trilobites reach their peak diversity, with estimates suggesting that more than 5,000 species evolved during this period alone. This radiation occurred as the continents continued their slow drift across the surface of the planet, and as sea levels remained high, maintaining the extensive shallow seas that provided ideal habitat for bottom-dwelling arthropods. Trilobites diversified into forms adapted to every conceivable marine environment, from the sunlit shallows of tropical reefs to the dark depths of the continental slopes, from muddy estuaries to clear open waters. During the Ordovician, trilobites evolved increasingly sophisticated eye structures, with some species developing the schizochroal eyes that would become characteristic of the phacopoid trilobites. These advanced compound eyes possessed fewer but much larger lenses than the holochroal eyes of earlier trilobites, and each lens was a complex doublet structure that corrected for optical aberrations, and provided exceptionally clear vision. The spaces between the lenses in schizochrol eyes were filled with calcite, creating a solid eye structure rather than the membrane-covered arrays of earlier forms. This innovation allowed these trilobites to see with remarkable clarity and may have provided advantages in predator detection or in the location of food resources, though the energetic cost of building and maintaining such complex eyes suggests that vision must have been extremely important to their survival strategy. The adaptations that trilobites evolved extended far beyond their eyes. Some Ordovician trilobites developed the ability to enroll completely, curling into a perfect ball with the head shield and tail shield fitting together so precisely that no soft tissues were exposed to potential predators. This defensive posture, preserved in countless fossils showing trilobites frozen in their protective curl, provided effective protection against crushing or biting attacks, and may have allowed trilobites to survive in environments where predation pressure was intense. Other trilobites evolved elaborate spines and tubercles on their exoskeletons, structures that would have made them difficult to swallow or handle, while still others became streamlined swimmers, reducing their armor in favor of speed and agility. The decline of trilobites began during the Devonian period, often called the Age of Fishes, when the radiation of jawed vertebrates created new predatory pressures that trilobite populations had never faced before. Fish with powerful jaws and crushing teeth could break through the calcite armor that had protected trilobites from invertebrate predators, and the mobility and sensory capabilities of these vertebrate predators made them formidable hunters in open water environments where trilobites had once moved relatively freely. Trilobite diversity began to fall during the Devonian, dropping from the thousands of species that had existed during the Ordovician to perhaps a few hundred by the end of the period. The groups that survived tended to be either highly specialized for specific environments where fish predation was less intense, such as deep water or low oxygen zones, or were generalists that could exploit a variety of habitats and food sources. The Carboniferous period, famous for its vast coal swamps and the first great forests on land, saw trilobites reduced to a remnant of their former glory. Only a handful of families persisted, occupying marginal habitats and ecological niches that the more successful groups of the time, including various mollusks, brachiopods, and the increasingly diverse arthropods like early crustaceans, had not fully exploited. The trilobites that survived into the Carboniferous were often small, inconspicuous organisms, very different from the large, heavily armored forms that had dominated Cambrian and Ordovician seas. They persisted not through dominance, but through their ability to survive in conditions that other organisms found challenging, demonstrating that evolutionary success sometimes means knowing when to retreat to the margins rather than competing directly with more recently evolved groups. The final chapter in the trilobite story unfolded during the Permian period, the last period of the Paleozoic era. By this time, trilobites had been reduced to just a few genera, restricted to specific geographic regions and ecological settings. The world they inhabited was very different from the one their Cambrian ancestors had known. The continents had assembled into the supercontinent Pangaea, reducing the total length of coastline and eliminating many of the shallow epicontinental seas that had once provided vast areas of suitable habitat. The climate was becoming more extreme, with ice ages affecting the southern portions of Pangaea while the interior of the supercontinent experienced arid conditions. Marine ecosystems were dominated by organisms that had evolved long after the Cambrian explosion, including reef-building corals, diverse mollusks and brachiopods, and increasingly abundant fish. The end came for trilobites during the Permian-Triassic mass extinction, the event that occurred approximately 252 million years ago and eliminated an estimated 90 to 96% of all marine species. This catastrophe, triggered by massive volcanic eruptions in what is now Siberia that released enormous quantities of greenhouse gases and caused global warming, ocean acidification, and widespread oxygen depletion, was too severe for the remaining trilobites to survive. The few genera that had persisted through the late Paleozoic went extinct along with countless other groups, and trilobites disappeared from the fossil record forever. Their 270 million year run had finally ended, making them one of the longest-lived groups in the history of complex life, though ultimately proving that no matter how successful or well-adapted a group might be, extinction is the ultimate fate of all lineages given enough time. The legacy of trilobites extends beyond their impressive longevity. They left us with one of the most complete and detailed fossil records of any group, with more than 20,000 species described from rocks around the world, and new species still being discovered regularly. This exceptional record has made trilobites invaluable for understanding evolution, biogeography, and the history of life on Earth. Because trilobites were widespread and diversified rapidly in response to environmental changes, they serve as excellent index fossils, helping geologists determine the age of rocks and reconstruct ancient environments. The different species and genera of trilobites that characterize different time periods and geographic regions provide a detailed chronicle of how life responded to the movement of continents, the rise and fall of sea levels, and the warming and cooling of global climate. Trilobites also demonstrate an important principle about evolution and adaptation. Their success was built on a body plan that proved remarkably flexible, capable of being modified in countless ways to suit different lifestyles and environments while maintaining the core features that defined the group. The three-lobed body, the calcite exoskeleton, the compound eyes, the biramous appendages, all of these fundamental characteristics persisted throughout the 270 million years of trilobite existence, even as superficial features like size, shape, ornamentation, and proportions varied enormously. This combination of conserved core features and variable peripheral features is a hallmark of successful evolutionary radiations, allowing a group to explore diverse ecological opportunities while maintaining the essential mechanisms and structures that made the original design successful. The trilobites of the Cambrian established this successful template, and their descendants carried it forward through almost inconceivable spans of time, adapting it to changing circumstances while never abandoning the fundamental architecture that their earliest ancestors had evolved during the explosion that birthed complex animal life. 541 million years separate us from the beginning of the Cambrian period, a span of time so vast that it defies meaningful comprehension. If we were to compress the entire history of complex animal life into a single year, with the Cambrian explosion occurring on January 1st, then the rise of the dinosaurs would not happen until late September, the extinction of those dinosaurs would occur in late October, and the entire history of human civilization, from the first agricultural settlements to the present day, would be compressed into the final seconds before midnight on December 31st. Yet despite this immense temporal distance, despite the hundreds of millions of years that have passed and the countless changes that have reshaped the surface of our planet, the basic patterns established during the Cambrian remain visible everywhere we look in the living world around us. Every major group of animals that exists today traces its origins to the Cambrian Explosion, a fact that becomes clear when we examine the body plans and fundamental anatomical features that define different phyla. The arthropods that dominate modern ecosystems, including insects, spiders, crustaceans, and their relatives, are descendants of the Cambrian arthropods that crawled across ancient seafloors and swam through primordial oceans. The diversity of insects alone, with more than 1 million described species and countless more awaiting discovery, represents an elaboration of the basic arthropod body plan that first appeared during the Cambrian. The mollusks, which include everything from snails and slugs to clams and oysters to the remarkably intelligent octopuses and squids, are the heirs of the shelled organisms that evolved biomineralization during the early Cambrian and used it to construct protective armor against the first predators. The echinoderms, that distinctive group that includes sea stars, sea urchins, sea cucumbers, and brittle stars, appeared late in the Cambrian as relatively simple organisms, but would go on to diversify extensively during subsequent periods, developing their characteristic five-fold radial symmetry and their unique water vascular system for locomotion and feeding. The brachiopods, those lamp shells that once dominated Paleozoic seafloors and still persist in modern oceans, though in much reduced numbers, established their basic body plan during the Cambrian and have maintained it with remarkably little modification for 500 million years. And the chordates, that small and seemingly insignificant group that included creatures like Picaia and Hycoichthys, gave rise to the vertebrates, which would eventually colonize not just the oceans but also the land and the air, producing fish, amphibians, reptiles, birds, and mammals. The innovations that emerged during the Cambrian were not limited to particular groups, but included fundamental capabilities that would prove essential to complex animal life regardless of taxonomic affiliation. Vision, which appeared independently in multiple lineages during the Cambrian, transformed the nature of ecological interactions and continues to be one of the primary senses through which animals gather information about their environment. The compound eyes of modern insects and crustaceans operate on the same basic principles as the eyes of Cambrian trilobites, using arrays of photoreceptive units to build images of the world. The camera eyes of vertebrates and cephalopods, though they evolved independently from very different ancestral structures, both trace their origins to the Cambrian period, when the first light-sensitive organs began to develop into true image-forming eyes. Biomineralization, the ability to construct hard parts from minerals extracted from the environment, revolutionized animal life during the Cambrian and remains fundamental to countless organisms today. The shells of modern mollusks are built using the same basic chemistry that their Cambrian ancestors employed, precipitating calcium carbonate in crystalline structures supported by organic matrices. The bones and teeth of vertebrates, though they use calcium phosphate rather than calcium carbonate, represent an elaboration of biomineralization strategies that first appeared during the Cambrian. Even organisms that do not themselves biomineralize are affected by this innovation, living in a world where shells and skeletons create three-dimensional habitat structures, where predators must contend with armored prey, and where the accumulation of mineralized remains creates vast deposits of limestone and other sedimentary rocks. The ecological patterns that characterize modern ecosystems also have their roots in the Cambrian. The existence of multiple trophic levels, with primary producers supporting herbivores that in turn support predators, was established during the Cambrian and has persisted as a fundamental organizing principle of life ever since. The arms races between predators and prey that drive so much evolutionary innovation today began during the Cambrian, when the first animals with eyes started hunting other animals, and their potential victims responded by evolving defensive adaptations. The partitioning of environments into distinct ecological niches, with different species specializing on different food sources, habitats, or lifestyles, emerged during the Cambrian as animals diversified and began to compete for resources. The basic structure of a functioning ecosystem, with its complex food webs and intricate interdependencies between species, was assembled during those crucial 55 million years. The Cambrian also established something less tangible but equally important: the evolutionary toolkits and developmental mechanisms that would allow life to continue innovating and adapting over the hundreds of millions of years that followed. The Hox genes and other developmental control genes that regulate how bodies are built appeared before the Cambrian began, But it was during the Cambrian that these genetic tools were deployed in the construction of diverse body plans and then refined through natural selection. The basic developmental processes that transform a single fertilized egg into a complex multicellular organism with differentiated tissues and specialized organ systems were worked out during the Cambrian, and they continue to operate in recognizable form in modern animals. When a human embryo develops, it passes through stages that echo our ancient chordate ancestry, forming pharyngeal arches that are developmental remnants of the gill slits that Cambrian chordates used for respiration and filter feeding. The scientific importance of the Cambrian extends beyond its role in the history of life to its continuing relevance for our understanding of how evolution works. The Cambrian explosion demonstrates that evolution can proceed at very different rates under different circumstances, with long periods of relative stasis punctuated by intervals of rapid innovation and diversification. This pattern, sometimes called punctuated equilibrium, has been documented in many groups and at many timescales, suggesting that the concentrated burst of change we see in the Cambrian represents not an exception to evolutionary rules, but an extreme example of processes that operate throughout the history of life. The Cambrian also shows us that evolutionary success is contingent on environmental conditions, that the same genetic variation that might be neutral or even disadvantageous in one setting can become highly beneficial when circumstances change, and that the trajectory of evolution depends on the sequence of events and the particular combination of conditions that happens to occur. Looking back across 500 million years of subsequent evolution, we can see how the foundations laid during the Cambrian have supported everything that came after. The fish that filled the Devonian seas were elaborations of the basic chordate body plan that appeared during the Cambrian. The first animals to colonize land during the Silurian and Devonian carried with them anatomical features and physiological capabilities inherited from their Cambrian ancestors. The dinosaurs that dominated terrestrial ecosystems for 165 million years were vertebrates, members of the chordate lineage that began in the Cambrian oceans. The mammals that survived the extinction of the dinosaurs and eventually gave rise to primates and through them to humans represent a continuation of that same ancient lineage. The birds that fill our skies today are descended from theropod dinosaurs, which were descended from earlier reptiles, which were descended from the first tetrapods, which were descended from lobe-finned fish, which were descended from more primitive fish, which were descended from the simple chordates of the Cambrian seas. This direct connection between the Cambrian and the present moment means that the innovations and events of that ancient period are not merely historical curiosities, but part of our own story. When you look at your hand, with its 5 fingers and opposable thumb, you are seeing a structure whose basic architecture was established when the first fish evolved paired fins during the hundreds of millions of years after the Cambrian. The bones inside those fins, which would eventually be modified into the limbs of tetrapods and then further modified into wings, flippers, hooves, and hands, represent an elaboration of the internal skeleton that vertebrates began developing in the Ordovician, building on the foundation of the notochord that appeared during the Cambrian. Your eyes, which allow you to read these words, evolved from the same ancestral light-sensitive organs that gave rise to the eyes of Cambrian chordates, refined over hundreds of millions of years into the remarkable optical instruments that can detect single photons and distinguish millions of colors. The oxygen you breathe enters your lungs and diffuses into your bloodstream through respiratory surfaces that are evolutionary descendants of the pharyngeal slits that Cambrian chordates used to extract oxygen from seawater. Your nervous system, with its central cord running along your back and its enlarged anterior end forming your brain, developed from the dorsal nerve cord that characterized the first chordates, modified and elaborated until it became capable of conscious thought, language, abstract reasoning, and the contemplation of its own evolutionary origins. Every aspect of your anatomy, every physiological system, every developmental process that built your body from a single cell can be traced back through an unbroken chain of descent to those simple swimming animals that appeared in the Cambrian seas and survived long enough to reproduce, passing their genes and their body plan forward through geological time. The Cambrian shows us that life, given the right conditions, will explore every possible variation, will innovate and experiment and try out new designs with a creativity that can seem almost purposeful, though it emerges from the entirely mechanical process of genetic variation and natural selection. It shows us that major transitions in the complexity and diversity of life can happen relatively quickly when multiple factors align, when environmental conditions favor rapid evolution and the genetic machinery is in place to generate variation in body plans and ecological strategies. It shows us that evolution does not proceed smoothly or predictably, but in fits and starts, with long periods of incremental change punctuated by shorter periods of dramatic transformation. And it shows us that we are not separate from this process, not observers standing outside the flow of life's history, but participants in it, products of the same ancient explosion that gave rise to trilobites and anomalocarids and all the strange and wonderful creatures whose fossils we find in the rocks of 500 million years ago. The story of the Cambrian is ultimately the story of possibility becoming reality, of potential being actualized through the intersection of chance, necessity, and deep time. For 3 billion years, life remained trapped in simplicity, unable to break through to the complexity that genes and chemistry made theoretically possible. And then, during a window that seems impossibly brief when measured against the full span of Earth's history, everything changed. The doors opened, the possibilities flooded through, and the world became populated with eyes and shells and nervous systems, and all the innovations that would define animal life from that moment forward. The creatures of the Cambrian seas are long gone, their soft tissues decayed, their hard parts transformed into rock. The oceans they swam through have been drained and filled and drained again as continents drifted and sea levels rose and fell. The mountains that bordered those ancient seas have been ground to dust, and new mountains have risen in different places. Every atom on the Earth's surface has been recycled countless times, passing through countless organisms and countless geological processes. But the patterns established during the Cambrian, the body plans and ecological relationships and evolutionary innovations that emerged during that brief and remarkable interval, those patterns persist. They surround us. They shape the living world we inhabit. They are written in our own bodies, in the structure of our skeletons and the organization of our nervous systems and the chemistry of the oxygen-carrying hemoglobin in our blood. 541 million years is a long time, long enough for continents to assemble and break apart multiple times, long enough for mountains to rise and erode away, long enough for asteroids to strike and ice ages to come and go and ecosystems to collapse and rebuild, long enough for trilobites to dominate the oceans and then slowly decline and finally go extinct, long enough for fish to evolve and colonize the seas, for plants and animals to move onto land, for forests to spread across the continents, for dinosaurs to rise and reign and fall, for mammals to diversify, for primates to appear, for consciousness to emerge, for a species to evolve that could look back across all that time and piece together the story of where it came from. And yet, despite all that time, despite all those changes, the Cambrian remains present in our world. Its legacy is not a museum piece, not something we can only access through fossils and reconstructions and scientific papers. Its legacy lives and breathes and moves all around us, in the beetles and butterflies and crabs, in the snails and clams and octopuses, in the sea stars and sea urchins, in the fish and frogs and birds and mammals, in every animal whose body plan was established during that ancient flowering of diversity. The Cambrian Explosion was not just the beginning of complex animal life, it was the foundation of the world we know, the moment when life's potential became manifest, when the basic architecture of animal existence was drawn up and tested and refined. Rest now, knowing that you are part of an unbroken chain of life that stretches back more than 500 million years, to a time when your ancestors were small swimming creatures in ancient seas, when eyes were first opening to see the world, when the first shells were forming to protect against the first predators. The patterns written in those Cambrian seas are written in you as well, in your DNA, in the structure of your body, in the very process of development that built you from a single cell. You are a continuation of that ancient story, a living connection to a time so distant it seems almost mythical, yet so present in every breath you take, every heartbeat, every moment of conscious awareness. The Cambrian teaches us that life is resilient, creative, and endlessly inventive, that complexity emerges when conditions allow it, that the foundations laid in the deep past continue to support everything that comes after, and that we are not separate from this grand procession of life through time, but intimately connected to it, inheritors of innovations that first appeared when the world was young and the future was wide open with possibility. Sleep well, descendant of the Cambrian seas. The explosion that began 541 million years ago continues in you. Good night.